So what are fire refugia? I apologize for this photo. These are the kinds of ecosystems, or kinds of savannas, if you like, that I have to work in. It's often difficult to see the pattern, but in this situation, we have a forest in this gap here and a savanna-like vegetation around it. And here I have a definition for fire refugia from Meddens et al., where fire refugia are landscape elements that remain unburned or minimally affected by fire, thereby supporting post-fire ecosystem function, biodiversity, and resilience to disturbances. And that's great, and that's worked well for us for a long time. But now let's think about what climate refugia are. So climate refugia are typically areas that are relatively stable or slower to change relative to surrounds that allows biota time to adapt and persist. And note here a focus on stability and biotic dependence, not on the actual state, which is a bit of an issue. So if we were to make an analogy between climate refugia and fire refugia, essentially our current definition of fire refugia would be like choosing desert or polar and saying that's a refuge and the rest is, bad news. And so it completely loses that biotic component, and it also doesn't focus on the stability component. I'm not saying that what we currently call fire refugia aren't fire refugia. I'm saying that those are fire-free refugia. I'll just make that caveat now before you get angry with me. And why do we focus on fire-free refugia only? Well, I think it's a colonial history thing. It's an issue that we've always focused on trees as a resource, and that's where we see the trees. So that must be a refuge, because the trees can be there. And it's the climax community that occurs without the disturbance and the like. But it really misses the issues of stability and biotic dependence. So biotic dependence, the question comes, refugia for what? I think if we look at what our species or ecosystems of interest require for their persistence, a lot of them require fire. In fact, fire-free areas are highly stressful or fatal for a lot of the flora of South Africa. So we have lots of these species or lineages that have these fire-related or fire-dependent life cycles, and if you were to put them in a fire-free environment or to switch fire off, they'd disappear. Just to drive that point home, this is the kind of 20 years of modus fires across southern Africa, and this is a modeled map of species richness across South Africa, and I think you'd agree there's quite a strong kind of overlap in the areas of high species richness and the areas of high fire. And in fact, I'd argue that more than half of our 21,000 species in South Africa are probably fire dependent or rely on fire in some way. Plant species, sorry. If we look at just the Cape Peninsula, so here we pulled together an inventory of the 2,697 vascular plants, the species that occur on the peninsula, and it turns out that over 2,000 of them require, or almost 80% of them require fire less than every 50 years for them to persist. So if you were to remove fire from that landscape, it would be lethal to by far the majority of the diversity there. In fact, most of the remaining 20% are fire tolerant. They just don't actually depend on fire for their persistence. So only about 3% actually require fire-free environments, and those are largely trees and a few succulents. So looking at that, you'd ask, well, why aren't we saying that fire-prone areas should be the fire refugia and areas without fire is the rest, right? That kind of actually makes a lot more sense than the way we currently use it. But either way, there's an in-between option. So looking at stability, well, there's variation among species and ecosystems and their fire preferences. So there's lots of different species or ecosystems that require slightly different but stable fire regimes. So we're not just talking about fire-free and fire-prone, we're talking about fire-free, slightly fire-free or rare fire, slightly more fire, high fire, different kinds of fire. And each of those should be considered, or could be considered a refugium in their own right. So here we have evidence of species dependence on different fire regimes, evidence of different ecosystems requiring different fire regimes. Here's some more where showing 3 or more distinct communities in mosaics, thank you, occurring in mosaics that require different fire environments. So we know there's variation in the fires that these lineages and species and communities require. So I then propose, okay, well, let's consider an expanded palette of fire-free and fire-prone refugia. Excuse me. where in this landscape, so here we have a map of the probability of fire, where in the top left there's very low probability of fire in the lighter area, and the darker red area is high probability of fire. And here you see a frequency distribution of the kind of numbers of pixels in each. And you can take you can cut that off at some threshold, say here, and you can say, okay, well, that's your fire-free environment, or what we would usually have called a fire refugia, what I'm calling a fire-free refugium. And this would be your fire-prone environment, or your fire-prone refugium. Of course, there's different ways to slice this. If you look at this, you can see it's trimodal, so you could actually say, well, maybe there's 3 different states, and you can draw up 3 different types of refugia. I think this kind of actually logically extends from the definition of fire refugia that occurs in most papers that talk about fire refugia, and the one from Medden in particular talked about unburned unburned or minimally affected by. And that minimally affected by is already stepping away from this idea of a fire-free refugium. So and people use it differently in every paper you come across. And lots of people are like, oh, our fire refugium, which only burns every 10 years, and our fire-prone environment that burns every year. So I think it's really just the idea of trying to formalize a concept that we are all quite comfortable with. We just don't use the right terminology. And it kind of changes the way you think about that environment. So I got to this point, and I thought, cool, yeah, that was very clever. I'm sure everyone will like this. And I thought I'll show it to a guru, which is always the worst thing to do. And he says, okay, well, how do your fire-prone refugia differ from fire regimes? And that was 3 years ago, and I'm still trying to work it out. But I think I've got the threads of working it out. One is actually by reading his book and going through his definition of fire regime. So he was talking about the frequency of fire, its intensity, seasonality, and spatial extent vary greatly in different ecosystems and climate zones across the world. These patterns of burning are called fire regimes, or pyromes at large spatial scales, and strongly influence the nature of plants that occur within a given regime. So right here, he's gone and said, okay, it's a fire regime if it's at a finer scale, and it's a pyrome if it's a larger scale. So I just did that and said, well, it's a fire refugia if it's at a much finer scale than a fire regime, and it's a pyrome if it's even bigger than that. So, got you, William. But obviously that's not satisfying enough. So you need a bit more than that. And I think the other major component is actually looking at stability. So if we look back at the definition of climate refugia, they're relatively stable conditions with particular climate regimes, or areas where climate is unlikely to show a dramatic shift in regime. So by that notion, then a fire refugia can be relatively stable conditions within particular fire regimes, or areas where fire is unlikely to show a dramatic shift in regime, right? So this is kind of narrowing it down. You now have your map of fire regime, but within that you have areas that are particularly stable, and because of that stability, you can end up with biotic dependence. So that's the trick, is that the predictions are that with stable fire-prone refugia, you should have areas with this biotic dependence on those stable refugia. And biotic dependence is a long list, but these are just the few of the obvious ones, things like endemic species with specific fire requirements, unique community composition, unique vegetation structure that might then support other biodiversity and the like. It's at this point where I was going to try and show you some very complicated analyses, and I decided I wasn't going to have enough time, so you'll have to wait for the paper or email me. I'm happy to share everything I've got. But what are the implications? Well, some of the implications are, I think it really helps us try and get towards a better defined meaning for biodiversity and the ways in which we measure biodiversity. Very few of the extant ways in which people measure biodiversity consider the stability component. And that's fine depending on the organisms you're working with. But essentially, relatively, if you consider the stability component, they're really important for sessile organisms. So most plants, I suppose. If it's a highly fragile organism like a bird, it doesn't really matter if everything's burning every now and then, as long as you have variation in habitat structure, perhaps, and provided they can survive the actual fire event. But when it comes to plants, if you have that random burning all over the place, that's seriously bad news if the plant can't get around. So for example, most fynbos species can migrate on average 1 meter, 1 meter a year, so you're not really going to get around fast enough to escape random fire regimes. So we really need to consider the stability component in the way in which we measure and think about biodiversity, but only dependent on the biodiversity that we're thinking about. So if it's highly fragile organisms, it doesn't really matter. If it's highly sessile organisms, then we really do need to think about it. And you'd predict that the highest diversity would be where you have a mosaic of different yet relatively stable fire regimes. Fire management, this is always a tough one because we kind of throw advice around, but we don't really think about what this means when it comes to actually implementing on the ground. This is the current fire management plan for Kruger, where they have 5 different zones with broadly defined thresholds of potential concern and guidelines. So it doesn't really consider this idea of some parts of this landscape might require more stable fire regimes at all. But the caveat there being that the park level guidance, this is my understanding of it, the park level guidance is very general, but then the managers in the different areas do have some flexibility to think about, like, it would be really silly to try and put a fire through this patch of forest on a koppie or that kind of thing. But it would be useful to think about and what science could contribute to the management if we were to think about where are there different kinds of fire refugia across the park. It's even more concerning if you look at Table Mountain National Park, where we manage it as one zone with one kind of set of TPCs. So I think that's where I'm working with the park quite a bit to try and work this out. And I'll leave that there. I just want to advertise 2 other conferences. If you got this far and really wanted to get to Cape Town, there's 2 other conferences in Cape Town this year. One is the International Statistical Ecology Conference in June, July, and then Medicos in September, the Mediterranean ecosystems. Thank you very much.
Thank you, Jasper. That was very good. There's a bit of time for some questions. Anybody out there? Sally, I knew you were stretching. That's not stretching. So, Sally, please.
Thanks, Jasper. That was a great talk.
Cool.
And I was thinking, aren't these just fire regimes? And then you answered my question. And I reckon you've nailed it. I like the scale stuff.
Thank you.
But I suppose I just feel like even with the climate stuff, there's this assumption that stability is good and what's needed by all organisms. It doesn't matter what fire regime they have as long as it's stable. But there are some instances where it's the variation at a point in space that allows the organism to persist.
Yeah.
Like a savanna tree needs a frequent fire regime, otherwise forest trees get in. But it also needs the occasional fire-free interval to become. So variability isn't always bad, right?
No, so that's exactly why I'm not showing you data, because I got into this kind of wormhole of trying to find the Goldilocks conditions for the different species. It is, as you say, a matter of rigid stability is almost always going to be bad news. So you need just the right conditions for a species where there's variability within some range. It's easy to come up with examples from fynbos where like too frequent fire means they haven't set seed, too long before a fire, and they senesce before a fire comes and the regeneration can happen. So there's a large amount of variability that would work for those species, but if you go too far over some or other threshold, then it starts to fall apart. If that makes sense? So yeah, too much stability is bad, but relatively stable. I did make sure I put relatively in front of every stable I put in the slides.
All right, one short little question here.
Thanks. To kind of follow on from Sally's idea, I mean, there's the Goldilocks condition for any given species, but in community ecology, the coexistence of different species often requires that things are shuffled around. What's good for one species isn't great for another, but if it's always good for that species, you end up just with sort of dominance and exclusion of other species, which I know is tricky with fynbos reseeders.
No, I mean, I think there's strong evidence for that in fynbos with this kind of spatio-temporal storage effect. And yeah, I completely agree that you do need that variability. I think that is actually like a big component behind driving, or where biodiversity comes in is a really interesting phenomenon, at least in that ecosystem. So this issue of things like block burning really freak me out, for example, because then you're making it, that whole area has the same fire regime each time, even if you're varying between your blocks. If you're not overlapping between them, then you're losing a massive dimension of diversity. I think I'm out of time. Sam's looking nervous.
Thank you very much, Jasper. That was a nice discussion there too, and good timing there. Our next one, we're going to now go back to animals, or start with animals. Andrew Davis is going to give us a bit of an update on some spatial heterogeneity and how carnivores coexist. Thanks.
Thanks, everyone. So yes, today I'm going to talk about how spatial heterogeneity facilitates carnivore coexistence. We hear a lot about spatial heterogeneity and biodiversity relationships. And the question we wanted to ask with this study was, does that apply to carnivore coexistence? And can we use it in this context? And as most of you know, lions and wild dogs are not good friends. And they, lions especially like to kill wild dogs, and they're actually the number one threat to wild dog survival and conservation, really. And what normally happens in most places of Africa is that wild dogs actually just avoid lions. So this is some data from a 2014 paper in the Serengeti. This is the very famous, lion study area within the Serengeti. And these little black dots represent wild dog GPS telemetry collars. And what you notice is there are maybe 2 locations out of many thousands within this area of high lion and hyena density. And so what normally happens is wild dogs simply run away from lions and avoid them as much as they can. However, that's becoming increasingly difficult for wild dogs. And this is an image from the Okavango Delta, a stronghold for wild dogs. And this is, you can see, human encroachment even in these kinds of systems. And so increasing pressure from outside of these conservation areas is forcing wild dogs and lions and many other carnivores to coexist and cannot actually avoid each other in space. But the Tlhuri Park presents an almost problem to this idea, because this is, as many of you know, a relatively moderately sized reserve in KwaZulu-Natal, about 90,000 hectares, and yet it's entirely fenced, I'll tell you that too, and it has very large growing human populations around most of its boundaries, and yet it has the highest wild dog density in Africa. It also has amongst the highest lion densities on the continent. And how does this actually work? If the wild dogs cannot avoid lions, they can't leave the park. How do they actually survive? And can we learn from this example in terms of carnivore coexistence as well as wild dog conservation? Because ultimately, wild dogs shouldn't really exist in Tlhuri. It doesn't actually make sense. So my hypothesis was that, for those of you who know HRP, it's a very spatially heterogeneous landscape. It has, these thick forest patches. It's got these open grasslands. It's very hilly. It's got a lot of spatial heterogeneity. And that's reinforced by these maps. This is the elevation across the park. The rainfall is highly variable across about a 30-kilometer distance. There's a range of vegetation types. And the question was, does this spatial heterogeneity, and the hypothesis was, does this actually facilitate carnivore coexistence? In other words, can wild dogs just simply hide from lions? And so what we did was we took overlapping GPS data from wild dogs and lions. We colored all the wild dog packs in the park, which are represented by these dots. Each color represents a different pack. And we colored every lion pride that we could get. We missed the lions in the wilderness area just simply due to logistics. And so there are some more wild dog packs down here that we weren't able, well, we didn't include in the analysis because we didn't have temporally matching lion data. But what this shows is this is a map on the background of lion density across the park. And you can see over here, for example, which is some of the highest lion density in HRP, and wild dogs don't seem to avoid it at all. So what we then did is we implemented an ISSA, which is an integrated step selection analysis. What this does is it allows you to include movement variables as well as habitat variables. So most movement ecology studies have really just consider where animals go in terms of habitat and landscape, but they don't really consider how they move differently in these landscapes. And ISSA incorporates both of those. So this black line over here represents where wild dog went, the observed steps, and the gray lines represent where it could have gone based on the distributions of its movement. We then looked at where these wild dogs were going over 2-hour time periods, which is why we had to constrain our analysis to only places where we had 2-hour data. And then we related that to where the lions were at the same time, where the lions were going, and we matched up the collars to within under about 2 minutes of each other, the fixes at least. We then mapped the habitat using LiDAR data, which I think most of you are familiar with, but this is just an example of what it would look like across the park, and this is a cross-section in one area. And then we thought, well, how do wild dogs use these landscapes in terms of vegetation? And to do that, we modeled view sheds. So basically, from every point where we had a lion GPS and a wild dog GPS fix, what can the wild dog or lion see from that point? And so you can imagine if you had a wild dog over here on this road, everything in this sort of lighter color, which represents sort of this light orange on the roads, and this sort of light green in the bush represents what the animal can see, and the darker colors represent what it can't see. So for example, you can see these trees over here occlude what's going on behind there. You can also see how you can, like, this is a dip over here, so if the animal was here, it could see across the dip to this area, but it couldn't see in front there. And so we modeled these view sheds to think about what can they see. Can they use this vegetation to hide or to see what's going on? We also thought about other variables. We included terrain ruggedness as well, not just vegetation, so how rugged are these areas. Lions, as you know, are very lazy creatures compared to wild dogs, so they don't really like to go in very steep hills or rugged areas. And then we also included time of day, because carnivores are obviously nocturnal and those kinds of things. So we broke this down between day, night, and twilight periods. And then we set up these hypotheses based on we constructed models based on these hypotheses. And we looked at 2 general types. We looked at whether wild dogs are simply avoiding lions. So basically, are they avoiding both lion proximity, which is symbolized by this, as well as lion encounter risk, so where lions are likely to be hunting. And if they were simply avoiding them, this is meant to be a dog running. It's kind of, yeah, anyway. Are they just simply avoiding lions like we normally see in most places? Or alternatively, in HRP, are they actually altering their use of habitat when lion risk is high? So we had these 2 lion risk categories again. So proximity would be where the lion is when the wild dog was there, based on GPS telemetry. Encounter risk was based on lion long-term use of the landscape, that utilization distribution map I showed you. And what do they do? In this case, the wild dog is not running, it's actually just more alert. So do they actually just know where the lions are and change their habitat, but don't actually try and get away from them? And if that's the case, then we'd expect them to use habitat differently, both vegetation and terrain. And so we set up these hypotheses, or set up these models to test these hypotheses, and we had various combinations of them to understand what wild dogs were essentially doing in clearing for lazy. Oh, and then we also included the different times of day, as I alluded to earlier. And the model that came out the strongest was this one, which is basically, it's a habitat encounter model, and that basically means that the wild dogs were adjusting their use of habitat when long-term lion encounter risk was high, which tells us firstly that they're proactively responding to lions. They're not waiting for a lion to be there. They're clever enough to know where lions should be, and they then adjust their habitat. They don't actually avoid these areas, they just adjust the way they use them. These are the variables that came in, so they increase their step length according to time of day and all these different variables, which means they go faster at different times of day, they go slower and other times of day. And the one that we want to focus on is this one down here, where they adjust their use of those view sheds I showed you based on how much lion risk there is and on the time of day. And if we unpack that a little bit, which was our main result really, this graph here, I'm going to show you a few versions of this, and what you need to know is that this is relative probability of use. So how likely are we to find a wild dog in this kind of area? This is lion encounter risk based on where they were over the period of the study. This represents 3 different levels of view shed. So areas that have a low view shed, the 10th percentile, are very dense areas. They're like thickets and those kinds of things where you could hide, versus the 90th percentile are really big, open, think about grazing lawns, that kind of thing. These dotted lines simply represent the percentiles same percentiles of lion encounter risk. In other words, 10% of the park is pretty low risk. More than half would be, or 90% would be up there. So this is during the day, we tell you over there. And you can see that basically most wild dogs, as lion encounter risk increases, there's a stronger chance of wild dogs being found in open areas, but it's not really significant, not really all that exciting. But down here, where lion risk is low, they tend to be in actually denser habitats. In twilight, they don't really seem to care where the lions are, and I think that's because wild dogs are hunting, so they're moving really fast, they're doing all sorts of things. Twilight refers to dawn and dusk. But then when you get to nighttime, what you realize is there's a strong shift, and the wild dogs are really, as lion encounter risk goes up, they are extremely selective for these very open areas and really unselective for these other ones. And what that's telling us is that wild dogs are actually not hiding from lions, but essentially in these open areas so that they can see where lions are coming from. And in HRP, wild dogs, I mean, in most places in Kruger, wild dogs are cursorial hunters that hunt down their prey. But in HRP, because it's so dense and they really feed mostly on Inyala, as well as Impala, they're actually using these thickets to hunt, it seems. And so they're not actually, their preference is actually for thicker vegetation, which is interesting in itself. When lions are around, they switch to these open habitats. But that's based on visual cues, we think. So what happens when you have dark nights? So we then compared this against full moon and new moon conditions, and you can see in full moon conditions, they basically do the same thing. There's even stronger selection for open areas where they can see lions coming. But in new moon conditions, this actually changes a lot. So in dark nights where there's no moon, lions actually, the model itself changed from a habitat encounter model to a lion proximity model. So what they then do is they're actually really, really worried about where lions are, and they change their response to actually be reactive to lion proximity rather than long-term lion risk. And they then, this model here, you'll notice, is ruggedness, not even view shed. They actually don't care about vegetation. And this graph over here, I'm pretty convinced is really just a statistical artifact, essentially that wild dogs are in rugged areas when the distance to lion is far, not because they're going to these areas far from lions, but actually because lions don't go to these areas. And so really rugged areas are simply because lions are not there. And what it really tells us is that they're not really altering their habitat use in terms of vegetation or ruggedness on these really dark nights. They're actually just trying to get away from lions, which is a more classic response that we could expect. So what does this tell us about wild dog-lion coexistence? Well, it tells us, firstly, that, the first point to realize is that wild dogs actually have evolved with lions. That's important to know because a lot of reintroduction efforts think about, well, let's get wild dogs into areas where lion densities are low. But that's not really realistic for starters. And secondly, lions and wild dogs have co-evolved. And so instead, like I said, it's unrealistic to have natural populations in the absence of lions. And I think what we should instead do is think about how spatial heterogeneity could actually provide a conservation mechanism for wild dog survival, and think about reintroducing wild dogs in heterogeneous areas. This study also shows us that spatial heterogeneity can be an alternative mechanism for carnivore coexistence in addition to temporal separation and spatial separation, as well as dietary niche separations. And if we think about open areas, this is Etosha National Park. Wild dogs were introduced to Etosha several times. They've gone extinct every time. They've gone extinct, locally extinct from the Serengeti Plains, from Lua Plain in Zambia. All these open areas like this where wild dogs actually don't do well. And we should actually think about this and rather focus on heterogeneous environments. And with that, I will thank these various people and point to you that the paper has been accepted. So if you want to argue with me, it's too late. But you can go and read the paper for more information. Thank you.
Thank you, Andrew. It doesn't mean they can't ask you a question yet. There's time for
No, of
one
course.
question. Brett?
Just taking what you're saying about the open habitat, would it be practical to introduce wild dog to Addo?
I mean, yes, it would, I think. I mean, Addo is like so thick that I wonder if that might be too thick, probably, for them to even get into that thicket. But ultimately, yes. I mean, I believe that wild dogs were historically present in Addo in the Eastern Cape. And so I would suggest, yes, it would be really interesting because of that thick vegetation. So I'd say, yeah.
Thank you, Andrew. I really appreciate that and a good story to tell. We're going to move on. I'm going to stay with carnivores with Kyle that's going to reflect a bit on temporal activity patterns. Thanks, Kyle.
Okay, good morning. I'm going to present a section of my master's research, which revolves around the activity patterns of carnivores. So when you take it down to the basics, the activity patterns of carnivores are dictated by light, so whether they are nocturnal, diurnal, or whether they are crepuscular, meaning active during dawn and dusk. However, there are a number of factors that may alter this, may alter their activity patterns. Examples being the seasons, wet and dry seasons, temperature extremes between winter and summer, and then weather events, social factors like reproductive behavior and mating periods, threats in the form of intra-guild predation and competition, and then obviously acquiring resources. So my data is camera trapping data that stems from the Snapshot project, and it includes Tswalo Kalahari, Madikwe Game Reserve, Klaseri Private Nature Reserve, and the other associated private nature reserves in the area, Mountain Zebra National Park, all in South Africa, and then Serengeti National Park in Tanzania. So what I did was I looked at the kernel densities of the photo captures for each species, especially the activity core, which is the top 50% of the densities. Also looked at the interspecific overlap and whether this may indicate temporal segregation and so avoidance. And then I compared this between wet and dry seasons, the warmest and coldest months of the year, and then obviously between the 5 sites as well. So this allowed me to include 21 carnivore species, ranging in body size from lions down to banded mongooses. So looking at each species separately and their temporal niches or temporal categorizations, lions, spotted hyenas, leopards, and brown hyenas were all considered to be predominantly nocturnal. And by predominantly, I mean that their core activity are within the nocturnal range. They do, however, have some activity during the daytime. As you can see with the graph here for leopards in the associated private nature reserves, the shaded area is the activity cores. And the x-axis ranges from 0 to 24 hours. I missed a few there. Cheetahs were considered predominantly diurnal, and African wild dogs were rather strictly crepuscular. Mesocarnivores, black-backed jackals, golden jackals, caracals, servals, honey badgers, aardwolves, and badgered foxes were all considered predominantly nocturnal, and then side-striped jackals and African civets were rather more crepuscular, more specifically nocturnal. For small carnivores, African wildcats, Cape foxes, genets, and white-tailed mongooses were strictly nocturnal. As you can see with white-tailed mongooses at the bottom left there, activity cores ranging around midnight. And then Egyptian mongooses and banded mongooses were strictly diurnal, with the bottom right graph for banded mongooses. Both of these graphs are for Serengeti National Park. When we look at seasonal variation for each species, a common trend that I saw was a reduction in activity in the morning hours during the colder periods of the year and also during the dry season, as you can see with the encircled areas. This is an example of brown hyenas within Madikwe National Park. And then another prominent example of what I just said is for aardwolves in Mountain Zebra National Park. When you look, for example, at the bottom right graph, there's no activity in the morning hours during your coldest months of the year, compared to almost all of the activity centered around dusk or just after dusk. Another common trend that I saw was an increase in activity during the daytime, especially for the nocturnal species. This is an example of black-backed jackals within Mountain Zebra National Park, and you can see in the encircled areas where you have, for the two graphs on the right, the dry season and coldest months, you have an increase in daytime activity. When we look at variation between sites, we see a prominent observation that we see is when you compare the 4 South African reserves with Serengeti National Park. And I think it's just because of the temperature extremes associated between winter and summer for the South African reserves. And Serengeti being so close to the equator, there's almost no difference. So as you can see, there's an example of mountain zebra in Mountain Zebra National Park when you compare with Serengeti for aardwolves. During the wet season, they have basically the same activity patterns. However, in the dry season, it's a lot different. Another interesting observation is for black-backed jackals, where with the 2 graphs at the top, which is Madikwe Game Reserve and Serengeti National Park, we have higher abundances of large predators. You see a more shift to a more crepuscular behavior. When you compare that to Tswalu and Mountain Zebra National Park, with lower large predator densities, you see the more classic nocturnal behavior. Another interesting observation was for lions in the associated private nature reserves and Madikwe Game Reserve, where in Madikwe they also shift to more crepuscular behavior, and in the associated private nature reserves they have the classic nocturnal behavior. And moving on to the interspecific overlap between species and looking at the classic lion and spotted hyena example, overall we see statistically different activity patterns for the 2 species. However, almost all of the differences and the variation are centered within the dry season. Again, in the wet season they are basically the same. As you can see here, the activity pattern graphs for lion and spotted hyena within the associated private nature reserves. And if you look at the 2 graphs on the right, which is the dry season and the coldest months of the year, it's almost a mirror image of the activity peaks for the 2 species. We have the activity peaks for lions within the morning hours during the colder periods of the year, and your activity peaks in the afternoons for spotted hyena, or just after dusk actually. I saw no differences in the activity patterns between leopard and lions. I did, however, see when comparing leopards and spotted hyenas. Because of the high nocturnality of African civets, it means that they do have statistically different activity patterns compared to the rest of the species, except for spotted hyenas. Because African wild dogs are so strictly crepuscular, and cheetahs being more active during diurnal hours, they have low overlap with the other larger carnivores, the more dominant carnivores. And so if they do employ a strategy of avoiding the larger and more dominant carnivores, that they can do so by temporal segregation of their daily activity patterns. Black-backed jackal had more overlap with lion compared to spotted hyena and brown hyena. And then spotted hyena and brown hyena again had statistically different activity patterns. But again, it is concentrated within the dry season. When the wet season, it's kind of basically the same. African wildcats show different activity patterns to mesocarnivores, but not to large predators. So the suppression of mesocarnivores on smaller carnivores like African wildcats is probably higher than the large predators. Looking at black-backed jackal's effect on aardwolf and bat-eared foxes, we see statistically significant differences in their activity patterns. In all of the reserves we had enough photo captures for them, which is the Mountain Zebra National Park and Tswalu, also in Serengeti as well. And the differences exist not only in the dry season, but the wet season as well, in the colder and hotter months of the year also. So we believe that if they do employ a strategy of avoiding black-backed jackals, that there is a possibility that they segregate through changing their activity patterns. We see the same pattern for brown hyenas with aardwolves and bat-eared foxes. However, this is again centered within the dry season, and we see no differences between black-backed jackal and caracal, which are 2 important mesocarnivores. For Cape foxes, we see a high overlap with possible intra-guild predators and competitors like black-backed jackal and bat-eared foxes. So if they do want to avoid the more dominant species, they do not use temporal segregation as a method to do so. So overall, we see a lot of variation within the results. We see that seasonal differences are very prominent within the activity patterns of carnivores. And whether the low overlap between carnivore species are indications of possible temporal avoidance in the form of active avoidance, we cannot say. We can, however, say that there is a possibility that they do use temporal measures to avoid the species. There's some results that support the idea that the activity patterns of carnivores are rather dictated by bottom-up forces rather than top-down suppression and regulation by more dominant predators. This is quite a large study that uses standardized methods, and this is beneficial because when you compare previous studies, similar previous studies that look at the activity patterns of carnivores, it's sometimes difficult to compare between the studies just because they use different methodologies of gathering data and also analyzing the data. So having 21 species over 5 rather distinct sites is quite beneficial. I also included a number of underrepresented species in the previous studies, such as white-tailed mongooses, African wildcats, Cape foxes, and that. I believe this could aid future research planning and management interventions. I'll skip a point there. I also show the intricacy and complexity with the activity patterns of carnivores, and therefore advise caution to generalizing a certain temporal categorization for a species in a certain area before you properly investigate. Yes, like I said, it could aid in future research planning and management interventions like physiological studies perhaps, when you look at, for example, maybe metabolic rates and body temperature and that. I think it also serves as a nice baseline study for future research in the same areas. And then knowing the activity patterns of carnivores could obviously aid in the monitoring the health and movement of these species. And also it could aid in formulating solutions for human-wildlife conflict situations. Thank you.
Thank you, Carl. A nice illustration there. We've got some time for a question. Carl, do you think there's an opportunity to use this kind of analysis that you've got here to get to sort of predictive space? Because, for example, the application in human-wildlife conflict, you know, very often we're just describing what is happening, but it's be much better if we can get to a space of actually predicting where things are likely to happen in future. How can one think about applying this kind of stuff that you're doing here in that context?
With future predictions?
Yes.
Yeah, one aspect that I wanted to look at was looking at climate changes and how that might influence the future changes in their activity. Concerning human-wildlife conflict, I'm not sure. Yeah, I don't know. Something to think about, I guess.
Here we go. That's your postdoc.
my PhD, maybe, yeah.
Thank you, Carl, and really appreciate that. Right, we're going to move a little bit to the things that carnivores eat. Sandra hasn't said a word to me this morning, so I'm not, he must be concentrating a lot. So thanks, Sandra.
Right, good morning, everyone. Before I actually begin my talk, am I in the right place? Before I actually begin my talk this morning, I'd just like to remind you all again about the open invitation to join the workshop and special issue that we're going to run this year on biodiversity informatics in South Africa. We're funded by NITIX, and we plan to host a workshop that highlights some of the challenges and opportunities in South Africa for biodiversity informatics. and then produce an open access special issue that can hopefully showcase some examples of the use of novel data sets, maybe highlight the plight of long-term monitoring programs in conservation areas and so on. It comes from a perspectives piece that we're hopefully going to submit later this week. It's got a really great spread of co-authors from all sorts of different institutes and academic facilities. So if you're interested in joining, please come and chat to me, email me, or there's an online form where you can express your interest. And no more adverts. All right, so this work that I'm going to present today is a work in progress with a number of co-authors and myself, and we look at the spatial temporal dynamics of herbivore meta-communities in African savannas. Is it in the wrong place? Okay. I knew it had to go down at some point. All right, so yeah, it was inspired by some work that we've done previously on the updating of the herbivore management plan in Kruger. And it's directly linked to the first objective over here, which is to identify areas of local concern that's associated with herbivores and to define the extent of those areas. So to do this, we first started by identifying key resource areas for different feeding gills. And to do that, we used aerial census data. I've already seen a lot of you have already used that in some of your talks, so you're probably familiar with it. And what we did is we took the herbivore species from there and we classified them according to the different feeding gills, grazers, browsers, and mixed feeders. That data has latitude, longitude points, and each of those data points has got the number of individual animals within that group and the specific herbivore species. We then added in an average body mass, kilograms of body mass for each individual, and we times that by the number of animals that were in the group. We did this because obviously one steenbok is not the same as one giraffe, or those are different feeding gills, but you get the point. So at the end of the day, we have this 2 by 2 kilometer grids with the biomass of animals within those 3 feeding gills. And from there, we delineated hotspots using kernel density estimates from that body mass index that we added. So here are some animations that show you the density estimates of that. And you can see how these patterns differ annually by feeding guild. And that's the interesting part, I think, the temporal component. And looking at an animation like this is great, but how do we actually quantify what's going on, not just spatially, but temporally as well? So for the actual report, and at a broader scale, what you can see here on this figure is just the combination of all of the different feeding gills together. So that is overall biomass density of herbivore species. And I mean, you can already see immediately the rivers are jumping out there. But then we took it a step further, and we took hotspots delineated from those kernel density estimates, and we just plotted those on a map, as you can see here, and delineated the different zones of preference for these different feeding gills. So the yellow, sorry, you can't read it there very well, but the yellow is basically the grazer hotspots. The magenta, which is like a purple, that one, that is browsers, and then the cyan, or that light bluey color, is the mixed feeders over here. And then the other colors are the combinations of those. So probably the most interesting of that is the black color over here, down there, which is the convergence of all the feeding gills together. So that's where browsers, mixed feeders, and grazers all come together. And it's isolated to an area down in Lower Sabi and Crocodile Bridge. But if you take those those gills and separate them out, the browser-dominated communities seem to dominate the knobthorn marula savannas and the mixed bush willow woodlands of the central region, and the grazer-dominated communities, the grassier basalts, which makes sense, in the Pretoria-Skop Sauerveld, as well as the alluvial plains up north, so those northern plains areas, which also makes sense, which is always great when the results actually make sense. The mixed feeder communities then are scattered across the park, but they are typically associated with riparian areas, or the major river systems. But the variability of these patterns are a lot more nuanced than these delineated hotspots that we pulled out. And there's a lot of interactions that are going on in the background. We've got individuals that are being exchanged between different habitat patches. Then you've got more complex interactions between networks of groups of species and guilds of species that are connected by habitats. And that's what we were interested to look at a little bit deeper. But it came down to then, how do we quantify a meta community? How do we quantify that structure? And because of, I know Gareth has had problems with the data, because of all sorts of interesting things going on with the data, we thought a nice way to, or an interesting way to look at this would be to look at the spatial dissimilarity of this herbivore biomass to help us delineate these zones of range convergence and transition. But then we still sit with the problem of what measurement do we use? There's a number of different dissimilarity measures that you can use. There's the obvious variability, dissimilarity, or diversity, or entropy. And entropy is used in a lot of different fields, including physics, ecology, even psychology. But it's essentially the variability of elements within a specific distribution. And probably the most obvious for all of you in the room has been Shannon entropy. That accounts for richness and relative abundance, in this case of herbivore biomass values, but it doesn't consider the magnitude of those values. And that's a big problem when you want to start comparing a biomass value of species gills. So we started experimenting with something called the Rouse quadratic entropy, and that takes into account this magnitude at a pairwise distance along with the richness and the relative abundance that the Shannon's index does. And it takes into account both the proportion of the cells, because this is then a spatial representation, assuming that different herbivore biomass values, as well as the degree of difference between those values, so the dissimilarity within that. And since we're using these kernel density estimates of the feeding gills themselves, at the end of the day, the higher Rao entropy value would represent sites whose surrounding biomass values are more diverse than the sites with a lower value. So the results then are a continuum of significant differences in gradients of herbivores biomass and are suggestive of potential meta-community gradients and boundaries. So an easier way to maybe explain that is with this little diagram here. And the essential difference between this and Shannon is that if you had a pixel that had a value of 2, and then you had one with a value of 1 in 100, and you can see that over here in this example where you've got low evenness and low diversity, and low evenness but high diversity. The values are exactly the same because it does not measure the difference between the 2 and the 3 versus a 0 and 100. There's a much bigger gap between those two values. And by adding in the Rouse quadratic entropy instead, we can capture that variability. So Shannon has a big problem with the evenness and the diversity measures and separating them. Okay, so we've done that then for all the kernel density estimates, and we've got then the annual results of these Rao quadratic entropies. The green areas essentially represent sites that are indicating there's a transition in the body mass, or the biomass rather, of the area, and the magnitude of that density is potentially illustrative of meta-community boundaries. So this is something we still want to test. And the patterns are looking really interesting, but I can't tell you what they mean right now. So if any one of you want to sit down and chat to me about it, I'm more than happy to do so. But we're going into it in more detail to look at what's going on. And those individual years, we can now actually use a locally weighted scatterplot smoothing function to calculate the average slope or the amount of the diversity along a temporal trend. So we can see how is that diversity changing within the landscape because we have this annual view of the entropy. And at the same time, we can create a temporal coefficient of variation, which is essentially the variability of those entropy values across the whole time series. So you'll get an overall map of turnover, basically. It's at the end of the day, it's represented by a percentage, and it's the ratio between the standard deviation and the mean of all those entropy values. And essentially, then the larger percentages represent a higher turnover in this biomass allocation in the landscape. So if we have a look at that, separated out into guilds. So this is the mean temporal diversity. This is summarizing essentially the slope and represents the amount of mean diversity along a temporal trend. So how is that diversity changing within the landscape over time? And also, once again, it's got some very interesting patterns. Making sense of them is, I'm going to sit down with people a lot smarter than me to try and have a look what's going on. Right now it looks like a Picasso image, but definitely getting some patterns for grazers up north and mixed feeders as well. But it makes a little bit more sense when we start looking at the coefficient of variation instead. So this is now representative of the temporal turnover, basically, of this spectral Rao entropy. And here you can see some patterns that are emerging for the browsers. Down here in this central region, the grazers seem to be down here in the lower Sabi area and along that boundary here. And the mixed feeders are doing a lot up north. So it makes some sense to me. I mean, when you look at the mixed feeders especially, they're going to be the species that are going to change more when the system is changing. And the north is an area, I think Tony showed us a picture in his talk, what it looked like up there in the dry season. So up there in the north, that is where animals are going to have to switch their diets quite quickly. So I would expect to see that the mixed feeders would probably have more changes in their, when I say changes in the biomass, obviously they're not getting skinnier. The big animals are moving and the small animals are moving out. So it's just changing that level of intensity of resource use, essentially. So where do we go to now? So we've gone through and we've delineated some of these range convergences and transition areas. We still have to look at, are they real and what do they mean? And to do that, we want to still look at some of the overlap. So what we did similarly with the hotspot analysis, we want to look at this on a continuum now, from what I've showed you here, and look at the overlap in these groups, but then also calculate some sort of level of co-occurrence, whether it's ranging from 100% co-occurring or 100% separated. And we will do this on all species. So I clumped them into these feeding guilds for now so that we could see if there were any interesting patterns that would emerge. But there are 26 species, so we're going to actually go through a process of looking at these ecological patterns and if there's synchronicity between the individual species and not just the guilds, which then would be quite exciting to look at when you've got known species that you know should have some synchronicity. And associated with that will be the temporal associations that I've shown you here, but the spatial, but then the temporal synchronicity is something that's going to be really, really interesting. And so the last would be to explore what is driving these patterns. And listening to the talks this last few days, some of you already answered some questions. I think Andrea was talking about the temperature, how that's affecting movements. Mika as well was talking about how temperature's changing. Sally was talking about how fire is driving the patterns of herbivores. And so if any of you want to collaborate further on this with us, please come and chat to me, and I'd love to hear your opinions. Thank you.
Thank you, Sandra. Your advertising and explaining a very complex row. I thought it was a dance of some sort, but anyway, it's not apparently. You're welcome to have a chat with Sandra and some of us who's working with her on this particular program and throw some ideas at us. Thank you very much. All right, we're going to move on to Boet Adrian Schrader, who will talk to us about olfactory landscapes.
Okay, thanks, Hams. So good morning, everyone. What I want to do today is something a little different than what I've done in the past at the Savannah Networking meeting. I want to talk about something a bit conceptual. And the ideas of incorporating olfaction into landscape use have been banging around in my head for a couple of years, and I've never been able to actually put them down or can put them together and flesh them out. And then COVID happened, and my PhD student, Pat, who's the first author on this paper, got trapped. He's from Australia, he's supposed to come out and work on elephants, couldn't get to do field work in Australia. And so I approached him and said, look, here, I've got these ideas, take them. And you'll see in the program that I talk about 2 Fs, and Pat has expanded this and turned this into 4 Fs. So he's now watching in Australia, sitting there with a beer, the cheeky bugger, and so hopefully I don't let him down. So if we talk about landscape use, basically we're talking in an aspect that has fascinated ecologists. We've seen it in a number of talks. Why do animals use this area? When are they there? And how long do they stay? And this really has kind of a range of uses. From my standpoint as a behavioral ecologist, I'm interested in animals and the decisions that they make. But there are very practical applications for conservation and management. And I think what we need to understand is that the interactions between animal and plants influence the structure, the dynamics, and the trajectory of populations, communities, and ecosystems. And we're talking about both animals and plants. And when we talk about that, this is generally where we start. So this is where most people here, this is food. This is the distribution of food and water across the landscape. This is the physical landscape. And what you'll find, there's been a lot of talks that have focused on this. And this is where they start and where they stop. But there's more to it. Animals don't sit there and say, well, okay, it's only the fact that, well, I'm a grazer, so I'll be in that grassy area, and browsers will be in the woody area. There's more to it than that. One thing that comes up, so this is the first F. The second F is fornication. Okay, so this is a family show, and it's on YouTube, so you can adjust that any way you want. But what you're looking is how animals are spread across the landscape in relation to breeding opportunities. Males establish territories. Females then, or leks, females then move across the landscape to look at males and have breeding, or weigh up breeding opportunities. Males can follow females across the landscape as well. The third F is fighting. And this is really interactions with conspecifics, and this can be aggressive interactions. This can be competition like scramble competition. The fact that you need food, there's other individuals of the same species, they're eating that food before you get there. There can be interference competition. You found food, they come and knock you out. Okay? You can also have situations where it's not just your own species, but other species, but it can be despotic distribution. The fact that males have established territories and you're not allowed there. So this is another aspect that comes in. Okay, so that's the third F. What's the fourth F? That's fear. We've seen this landscape of fear concept that overlays, that looks at physical landscape features here and relates perceived predation risk. Animals say, well, woodlands are scary. Don't go into woodlands because that's where the leopards are. Okay? And so we see a distribution of animals across that. Now this seems obvious, and most of you are saying, yeah, well, I completely understand that. But please find me the studies that incorporate all these aspects, because they don't. We pick a couple and then we ignore the rest. Okay? So if we take a look then at these 4 Fs, we see that really spatial use of herbivores is dictated by the trade-offs between the risks and reward. So first we can talk about, and I've just highlighted some really cool papers here. Scott Creel's work in Yellowstone, where he talks about the elk and wolves. And what happens is wolves move into area, elk push close to woodlands, they feed there, they get lower intake. One of my favorite papers is this Bonnier paper here, looking at zebra in Hwange National Park. Lions move in, the zebra can move completely out to move into horrible grazing areas, and the nutritional gain tanks. So they suffer nutritional loss to avoid predation risk. Next one, to get away from predators, let's talk about Scott Creel's work. And maybe you've seen this, you know, the fantastic thing where he's got elephants that have satellite collars, and then this little patch greens up. They use NDVI, and the elephants go yoop, and they move over there. And then this patch greens up over there, and the elephants go yoop. Okay? And it's really cool. It's really neat patterns. But the one thing that's bothered me about all of these studies, and I'm not attacking these studies in any way, shape, or form, how do the animals know? How do the animals know that the lions have moved in? How do the animals know that 5 kilometers down the road it's green and food's available? Okay? And so this is one of the things that we start getting into, the idea of animal senses and how they process information and utilize that information to determine how they move across the landscape. And one of the things that you'll find is this is hardly explained. You get these kind of random throwaway sentences and discussions. Well, the animals, may have seen it. And I don't want to, like I said, I'm not belittling these papers, but it's this idea of information that's vital. Things that come up, they talk about visual cues, they'll talk about auditory signals, and they may even talk about spatial memory. Now, the one thing is all three of these have flaws at some time or another, okay? If we take a look at visual cues, you take diurnal animals, it comes night, the vision drops off. Even nocturnal animals have to deal with thick vegetation blocking views. Like if you take a look at this picture, it's kind of find the leopard. There it is. It's behind me, oddly enough. Yeah. But the other thing you have is you have auditory signals. People say, well, lions move into an area, they mark their territory by roaring. You have elk that bugle to signal their body condition. But the problem with auditory signals are, as soon as you stop talking, the signals are gone. They don't linger across the environment. And last time I checked, predators don't go, okay, everyone, I'm just going to let you know I'm coming out, I'm hunting now. They don't talk. So they keep those sounds quite silent. Spatial memory is fantastic in pretty stable environments. But in arid regions where rainfall is unpredictable, spatially and temporally, how do animals move to water? Okay. So these are ideas that we've got. And also moving into novel areas. How do animals do this? And one of the things that I've been working on, you probably have noticed over the years of my presentations here, is that odor. And odor is one of those things that we tend to forget, all right? And the key thing about odor is that it is everywhere, and it lingers in the environment as a signal, okay? And the one thing about it is that it is a constant, ever-updating source of information for animals. Think about this. This is the Google Maps of how you get across the landscape. Right? So I live in Pretoria, it's a big city, I gotta get to work, I check Google Maps, okay, don't go down the highway, there's an accident. Well, this is what animals use. This ever-shifting land of information, ever-updating, always up to date, okay? It can be deliberately put there, things like scent marking, rhinos going along, spray urinating, or any territorial male spraying their territories. Signaling sexual states. You remember Courtney Monowick, one of my PhD students, and her work on olfactory communication in white rhinos through using dung. It can be incidental. It can be these volatile organic compounds that are given off by vegetation quality. Joanne spoke about it on Monday. You remember maybe Melissa Schmidt's work that she did with me as a PhD student on elephants using olfactory cues to decide what and where to eat. It can even be bacteria in water. 2 years ago, Doug presented his work on elephants smelling water. It just came out 2 days ago in the latest issue of Animal Cognition. So if we then go back to these 4 Fs, this information that's there, or what drives information use, you can see that every single one of these provides odor information. And so I'm proposing an odor landscape that sits on top of that physical landscape, that shifts, that moves, that animals use to determine where to be and what. So find out where food is, find out where conspecifics are, who to avoid, predation risk. So if they tap into this odor thing, they have basically a way to make efficient decision making, which is vital, okay, to get across landscapes. We see this by incorporating an olfactory landscape into behavior and movement frameworks. We start to provide a mechanistic link of how animals move across. We start to answer questions, How do they locate patches? Right, there's tons of information, tons of theory about patch departure rules, marginal value theorem, Joel Brown's giving up densities idea. But how do they find them in the first place? How do they decide, okay, I'm going to go over there and eat? Also, how do they know when it's safe to visit? We've all done this with landscapes of fear. You go, all right, these are scary areas of the landscape. You'll never find herbivores there. There's high risk, and then there's always those individuals that don't read the memo, and there they are in the scary area. How do they know to be there? Well, maybe they can smell that there aren't predators there at the moment. So it may be scary if predators are there, but if the predators aren't there, it's fine to be there. What we then see is that if we understand and utilize and come to grips with an olfactory landscape, we can modify this odor landscape, and we can use it for management ideas. So one that was done a while ago, Wayne Linkletter put the idea of putting black rhino dung from those individuals that are going to be released, put them out in the environment to try and reduce aggression from residents. Okay? You can also have a situation of protecting vegetation. So this is some of Pat's work that he's doing by creating associational refuges for plants. By using odor of non-palatable plants, close to palatable plants, animals may avoid or take a long time to find those plants. And this is great in restoration work. You could possibly use this for crop protection to reduce human-wildlife conflict. And also in our ever-shrinking, ever-fragmenting landscapes, we're doing things. We're building corridors, we're expanding networks to link game reserves. But the question is, how do the animals know where these bridges are? They don't move quickly into these areas once you open them. And especially, like, you take this is really cool. So this is in Canada. They've built these vegetative corridors across highways so they don't get blat by buses, all right? And so what they do, the question is that, well, how do they get from here to here, or vice versa? How do they know where that little corridor is? Well, maybe we put some dung or urine of conspecifics across that, and they see it's a safe place to be. So why am I doing this? Why am I presenting this here? My hope is to get people to start thinking bigger, going beyond just one of those F's and expanding and incorporating all of these, and of course, the olfactory landscape. My hope is that this stimulates new ideas with you, within your research groups, and how you perceive some of the data that you have. Why did those animals go there, but they didn't go there? And thinking beyond, well, it just must be random, or they didn't find it, or things like this. It's also to kind of start fostering novel approaches, especially when it comes to looking at management. If we can start to manipulate odor landscapes, we can start to have greater impacts and less hands-on impacts to shift animals across landscapes, okay? And get them in areas where we want them to be, in areas where they shouldn't be, whether it's poaching pressure or what have you. So with that, what I want to do is say that if any of these ideas have stimulated you, if you thought, wow, that's really cool, or you want to be part of this, please come talk to me. I've got my email address up there. We're doing a lot of things, you'll notice, and unlike Andrew, our paper is still in review, so you can argue with me. But my hope is that what you'll do is you'll walk out of here, and you won't open your eyes to new opportunities. You'll take that big sniff, and you'll smell the future of your research. Thanks very much.
Thank you, Adrian. Just tell them what deodorant are you wearing so they can sniff you out.
None.
We've got time for some questions.
Thanks, Adrian. I mean, I don't know, anybody who owns a beagle will shake their heads. We know very well that animals smell and sniff and follow it. I've got my question now. No, I'm just wondering, maybe the blindness, as it were, to the olfactory landscape is because we've got good tools for the visual stuff. We can model it, we can detect it with our own eyes, the auditory as well. But we're a bit challenged ourselves with smell, and we don't have good tools for measuring it. I mean, do you think maybe that's why there's this gap in the literature?
Look, Susie, I think basically it's the fact that we tend to focus on our own way that we perceive the landscape. And one of the biggest challenges in behavioral ecology is to put yourself in the animal's perspective. What you need, and what everyone needs, is a tame chemical ecologist. They're amazing. because they have the equipment to do this. I mean, some of it, yes, the technology is quite bad. We were talking about with white rhinos kicking up dung, that I'd love to be able to just hold like an air pump over it, suck in those compounds and see if that spreads their scent further. And the technology, it just can't handle it, but it's getting better. But still there are ways to do it. So we are doing it, we have been doing it. But yeah, you're 100% correct. We focus our lives on visual, like that, as opposed to an olfactory landscape, which, yeah, your dogs will take a dog for a walk and you know exactly what I mean.
Thank you, Adrian. That is an interesting perspective and a very good few ideas. I think a lot of people will go and smell him out. I'm also smelling the fear of some presenters to come up here. So, our next one is a short 5-minute presentation by Natalie on mammal road use. Thanks, Natalie.
Hi, good morning. So my talk has nothing to do with fornication, but it's hopefully still exciting. So the research that I'm presenting this morning was mostly conducted by one of my students, Misha Malarbe. There she is. But Misha couldn't be here herself, so hence I'm presenting on her behalf. So the idea behind the research originated a few years ago when we were driving around in Kruger, and I started wondering whether there's a difference in game viewing and game spotting between tar roads and dirt roads. We know from the literature that roads often have negative ecological impacts, and there are even studies that compare tar roads and dirt roads, and studies that have found that tar roads have a more negative impact, mostly because of the traffic volume, but also because they're broader, for example. So the most relevant study to our research is probably this one from Journal of Zoology in 2016, where they specifically found for impala that the tolerance distance is increased next to tar roads. In other words, they don't come as close, but they also found fewer impala sightings next to tar than dirt roads. Not that it makes a big difference, because you obviously see impala everywhere, but we then thought, well, this should be easy to test, not just for impala, but for all of the commonly spotted mammal species that you see often enough to actually be able to say something meaningful. So we did this using 3 hypotheses. We first hypothesized that there would be fewer sightings next to tar than dirt roads, that there'd be smaller herd sizes next to tar than dirt roads, and fewer close proximity sightings next to tar than dirt roads. All of this based on this increased disturbance that many papers describe from tar roads, mostly because of the increased traffic volume. So we sampled all the way from Punda Maria in the north to Malalan in the south, along the main tar road connecting, running north-south, as well as equivalent dirt road stretches that ran parallel as far as possible. We stopped every kilometer for 10 minutes, recorded all of the mammal species that we saw, estimated the distance to the closest individual to the vehicle, as well as herd size, and just to sort of confirm traffic volume, we also counted cars. There were slightly more tar road stops than dirt road stops, but there wasn't a huge difference. And then we did some stats to test for associations and for relationships. Okay, so what did Misha find? Well, firstly, it really doesn't make a difference. So you are equally likely to spot at least the 7 most commonly occurring mammal species, or that we spotted most often, from tar and dirt roads. So these are the results from chi-square tests of association, and what we see here are the presences from those 401 tar road stops and the 369 dirt road stops for the 7 most cited, that we cited most often, mammal species. First for tar roads and then for dirt roads for each of the species. And because the sample sizes were so similar, you really don't even have to look at the p-values. You can actually just get a feel for whether there's a difference by looking at these values, and they really couldn't be more similar in my opinion. So it really doesn't make a difference for these species in terms of game spotting at least. Then when we look at herd size and distance to the road, these are results for the three most that we spotted most often, elephant, impala, and zebra. First for herd size and then for distance to the road. Again, we don't really see a difference. The only one that sort of suggests that there's something there was elephant, with the data sort of suggesting that you're more likely to view elephants close up next to tar roads than dirt roads, but it really wasn't a very clear trend. So that's why I put a question mark down there. Yeah, so in general, what the data confirm is what we also see in the park, is that species are habituated, but more specifically that they're equally habituated to the different road types, to tar roads and dirt roads. And because we saw this for all 7 of the species that we could actually say something meaningful about because they occur often enough, it sort of suggests that it's possibly the case for less sighted species. From an application perspective, if you think at the moment, all of the dirt road closures because of maintenance and rainfall, this shouldn't really make a difference just from a game spotting perspective, because you are probably equally likely to spot that leopard from either road type. Thank you.
Thank you, Natalia, and I'm sure you can have a few discussions with people during tea time out. Thanks. Okay. All right, our next discussion is on the survival of white rhino calves, and Christoffel, or Stoffie, I think is what Adrian calls him. He's going to take us through this for the next 50 minutes. Thanks, Stoffie.
All right, so white rhino numbers are declining fast, and ensuring white rhino calf survival could be critical to conserving the species. Unfortunately, within Itala Game Reserve, nearly a quarter of all the calves born between 1999 and 2019 have died, and nobody knows why. When we actually started looking at this data, we very quickly realized, and as you can see from this graph, where the blue line indicates rainfall and the orange bars indicates the calf deaths, most of these deaths were occurring within the dry season or just afterwards, right? And no deaths were occurring during the peak wet season months of December, January, and February. Now, while this did not come as a surprise to us, it did give us an indication into what was actually causing these deaths: seasonal rainfall. You see, within African savannas, seasonal rainfall often influences the survival of grazers by determining the availability and the quality of resources. All right, during the wet season, grass growth peaks, and rainfall triggers this production of an abundance of high-quality food. But during the dry season, grass growth stops. Animals must endure this absence or shortage of high-quality food until the end of the dry season. But dry season rainfall triggers grass regrowth and therefore lessens the severity of the dry season. We also understand that the effects of dry season rainfall, or the effects of rainfall on survival, can actually vary depending on the availability of key resource areas. The reason for this being, key resource areas offer reserve and buffer resources that animals can rely on when nutritious forage declines, right, thereby modify this effect. So just some definitions for you guys. Reserve resources, those are types of resources of adequate quality that herbivores can rely on to support their survival during the dry season. Whereas buffer resources are those of a poorer quality that they can use to alleviate malnourishment in the absence of reserve resources. Now, white rhinos have a very strong seasonal use of key resource areas. All right, during the wet season, they prefer to feed in these highly nutritious short grass grazing lawns, but at the start of the dry season, these lawns, grass growth in these lawns stop, and instead they shift their foraging to reserve resources within woodlands. All right, but as the dry season progresses, eventually the woodland grasses senesce and brown, and they start supplementing their diet with buffer resources from tall grass bunch grasslands. So our aim of this study was basically to determine what was causing these deaths within Itala by investigating how different rainfall parameters affected the likelihood of calf survival, and whether these relationships were actually modified by the availability of key resource areas, being woodlands and bunch grasslands. So we predicted that dry season rainfall will positively affect the likelihood of calf survival by maintaining reserve resources throughout the dry season. We also predicted that an increase in woodland availability would strengthen this effect because it increases the amount of reserve resources being maintained. Then, we also predicted that dry season duration would negatively impact calf survival simply because it determines the time that calves have to endure low-quality diets, and that an increase in bunch grassland will mitigate this effect by offering buffer resources. So to do this, we use sighting data collected by Itala staff between 1999 and 2019 to gather information about all the calves. We then use the sighting locations and a habitat map of Itala to determine the percentage of each home range that consisted of woodlands and bunch grasslands. We then use this as a proxy to view the availability of each key resource area. However, when we did start taking a look at this, we found that bunch grasslands and percentage woodlands had a very strong negative correlation. So what we did, we removed bunch grasslands from the analysis and we focused on the percentage of woodlands. But I just want you to bear in mind this trade-off for later in the presentation. All right, it's very important. We then ran a logistic regression and a model selection process to see which variables were affecting calf survival. The variables that we included were dry season rainfall and duration. We also included the preceding wet season's rainfall and duration. The reason for this being, that is what determines the amount of food going into the dry season, as well as the amount of food available to the mothers during gestation. And then we included each variable's interaction with the availability of woodlands. So when we actually started looking at the deaths, all right, we first looked at the age at which the calves were dying. And we found that most of the calves were dying during their first dry season. In fact, they were dying during the first dry season, but never before the age of 4 months. Now, this is when calves still nurse predominantly, all right? So basically what this suggests is that despite nutritional stress, the mothers could still provide high-quality milk to these calves. Instead, most of the deaths occurred between the ages of 4 and 8 months, right? This is critical because this is the time in which calves begin to supplement most of their milk intake with grass, but they're still too small to actually tolerate declines in grass quality. Right, very few calves actually died older, and even fewer calves died during their second or third dry seasons. In fact, within our study, all the calves that died during their second or third dry seasons died during the dry season of 2002. All right, the reason for this being 2002 had a very long dry season of about 9 months. All right, that is a very tough time to be a white rhino calf. But now I just want to put emphasis on that 9-month dry season. All right, I've got a bit of a surprise for you at the end of this presentation. So we found that, as we had expected, dry season rainfall positively affected the likelihood of calf surviving, and that this positive effect was strengthened as the availability of woodlands increased, all right, eventually becoming significant once more than 66% of the home range comprised woodlands. Basically, all that this means is that when you have a high amount of rainfall, a high amount of woodlands, you've got an abundance of this nice green Panica maximum but reserve resources within woodlands. We also found that, as expected, dry season duration negatively affected the likelihood of a calf surviving its first dry season, and that this effect was exasperated as the percentage of woodland within a home range increased, all right, eventually becoming significant once more than 67% of the home range consisted of woodlands. Now, the reason for this being is that trade-off that I told you to remember, all right? Once the percentage of woodlands within a home range increases, the amount of buffer resources decreases in bunch grasslands, all right? All that means is that your calf becomes more and more susceptible to dry season duration's effect. Now, you might be wondering, how do these relationships actually affect calves within Itala? All right, To investigate this, we divided the calf population into 3 groups, some that had a low availability of woodlands, an intermediate availability of woodlands, and a high availability of woodlands. All right? And as you can see here, we have survival probability on the y-axis, the dry season rainfall on the x-axis, and then the dry season durations as the different types of lines. Calves that had a low availability of woodlands, there was no difference between their survival probability between 3, 6, and 9-month dry seasons. Right? Similarly, there was no difference between low rainfall dry seasons and high rainfall dry seasons. Right? So basically, these calves were buffered from these effects. However, I do want you to notice, if you look at this line represents the average amount of rainfall, dry season rainfall that Itola receives, which is about 80 millimeters. Despite being buffered, these calves only usually will have about 65% chance of survival. All right, that's not great. It's nice, but it's not great. But now, do note what happens when the availability of woodlands within the home range increases. All right, that looks very different. All right, so once again, here we've got an intermediate availability of woodlands, and here we have the high availability of woodlands within the home range. Right, survival probability in the y-axis and dry season rainfall on the x-axis. All right, so what we notice here is that during very short 3-month dry seasons, right, calf survival is extremely likely, irrespective of the amount of rainfall received. All right? In fact, if you take a look at the average amount of rainfall, you're probably going to have just under 100% survival, and over here it's about 100% survival. Right? That's great. But now let's take a look at what happens when the dry season duration increases. Okay, so this is your average typical 6-month dry season. You can see the survival probability when you receive the average amount of rainfall. It decreases a bit, but it's still quite high. Over here, it's still about 80%. Right, but what I do want to bring to your attention is that if you receive above an average amount of rainfall during your typical 6-month dry season, all right, calf survival is quite high. But if you receive less than average rainfall, right, you've got the sharp decline in calf survival. Right, basically what this indicates is that irrespective of your dry season duration, even your typical 6-month dry season, if that calf experiences a dry spell, a severe dry spell during that dry season, it could result in calf deaths. All right? But now, remember I told you to remember that 9-month dry season. Look at this. So basically, if you still receive the average amount of dry season rainfall that Itola receives, now you've got a calf survival of less than 20% and less than 10%. Right? In fact, if you want to increase your calf survival all the way back to where it's nice and high, Itola basically needs to receive double its usual amount of rainfall. All right? And that does not happen very often. So basically, I feel that is what is causing the calf deaths within Itola. Firstly, the calves that are experiencing dry spells, and it's the calves that are experiencing these extremely long drought-like dry seasons. So just to summarize everything for you guys quickly. All right. First thing we noticed was that the calves were dying during the first dry season. Right, this is when calves are still small. They require a high amount of high-quality forage, but they can't tolerate declines in forage quality. So the chances of them surviving during that first dry season was determined, well, positively affected by dry season rainfall and negatively affected by the dry season's duration. But these effects were linked to the availability of woodlands, to that trade-off between having reserve resources and having buffer resources. All right? Calves that had a low availability of woodlands, all right, they had enough buffer resources to be buffered against the effects of dry season duration, so there was little difference in their chances of survival. However, if you increase the availability of woodlands within a mother's home range, right now you are decreasing the amount of buffer resources within that home range. And now suddenly, you find that the calves are very vulnerable to, they are very vulnerable to dry spells and droughts. All right. Now, just to bring this to the broader perspective, right? These results raise various concerns with regards to climate change. All right, throughout Southern Africa, it is predicted that we will experience a decrease in dry season rainfall and an increase in dry season duration. Right, what that does mean is that throughout this region, we are likely to experience a very, very strong decline in white rhino calf survival. All right, so what our research does, it signals a warning, and it highlights some of the future challenges that white rhino calf survival could face within the very near future. Thank you very much. I appreciate you guys attending.
Thank you, Stoffie. I've got time for one question. Jasper?
So you looked at woodlands, but you didn't mention anything about grazing lawns. I was wondering how prolific grazing lawns are in that area, or if they're there at all, and if they're not, if there may be a potential buffering mechanism that managers could consider.
All right, so grazing lawns, well, Itala consists of about 2% of grazing lawns, so it's very small. And also grazing lawns require quite a lot of rainfall to stimulate growth. So we feel that during the dry season, they won't really play a role. It will more be the reserve and buffer resources within your woodlands and in your tall bunch grasslands.
Thank you, Stoffie, and I'm sure you're going to have a few more discussions with him over tea time. Right, we've got a short little 5-minute presentation by Lorenzo on predicting some spatial stuff for white rhinos.
So, thank you very much. Hello, everyone. So, as most of you may know, white rhinos are labeled as near threatened by the IUCN. Is it? Yeah, okay. Thank you. Okay. By the IUCN, even though since 2013, numbers in Kruger National Park decreased significantly. Because of that, we aim to provide a mathematical model to the managers of the park that can predict white rhino's movement response to environmental variables. And on your left, I think, we can see the data collection of some 30 individuals that were collected, the color data of some 30 individuals that were collected from early 2016 to late 2018. And on your right, you can see the visual representation of step selection functions, which our model will be based on. So how does it work? Basically, we take the location given by the color data, and for each location, we generate a fixed number of random steps. These random steps have their lengths are extracted by this real step length distribution. And we do the same for the turning angles. So after that, the step selection functions assign values of 1 to the used location and values of 0 to the randomly generated ones. And after that, we weight the environmental variables that are taken into consideration. And by doing that, we can estimate the probability of an individual to choose a location over another. So these are some of the different environmental variables we have taken into consideration. And lastly, we can see some preliminary results for 2 different individuals. So first, you can see that there are different variables for each individual, and this is due to the fact that we excluded variables that presented a high value of VIF because of strong collinearity. And most of all, we can see some of the significant ones, such as the elevation for both the individuals. So one has a negative coefficient, which means that this female probably, on average, preferred to go downwards, while the male preferred upwards landscapes. And for the distance to the river, for example, both of them were trying to avoid that. So this result may look strange, and kind of it is, but we have to take into consideration the length of the period of time, and this is an average on the period of time, and the concordance. The concordance actually is quite nice because it's larger than 0.5, which means that we actually have some hints that these are not randomly chosen. But still, we still need to implement a couple of variables, such as the time of the day and seasonality, and so that we can push the concordance level, which ranges from 0 to 1, as much as possible to 1, and then we can actually validate it. So thank you very much for your attention, and if you have any questions, you can either send me a mail or catch me at the tea.
Thank you, Lorenzo, and I'm sure you'll have a nice discussion with some of those anomalies. Males want to walk uphill and females downhill. Thanks, Lorenzo. We've got another 5-minute talk here by Catherine. She predicted that load shedding is going to happen in her talk, so I'm hoping for her that it doesn't. Thanks, Catherine.
Hi, everyone. Adrian set me up quite nicely to explain to you all why I'm so interested in trying to map rhino middens. And as he pointed out, rhino middens are these communal defecation sites that are really key to olfactory communication for both black and white rhino, and ecology like movement and behavior, and also can support some of the conservation management applications Adrian also pointed out. But in order to do this, we need to be able to map middens, which hasn't been done before. So we asked ourselves if we could efficiently detect rhino middens using high-resolution UAV imagery, and our preliminary results suggest this one on the lower left, but overall they were pretty difficult to find in such a large and heterogeneous search space. This image on the right actually has 2 middens in it that you probably can't see unless I point them out to you. But we're very spoiled in the Davies lab, and we also have high-resolution thermal imagery, and when we started looking for middens here, we saw that they appeared as these bright spots across the thermal landscape. So with this RGB image that we had in the last slide, if we overlay it with our thermal, we can see that those 2 middens really stand out as bright spots across this landscape. So we had these 2 great data sets that were collected by the Harvard Animal Landscape Observatory flown here at Kruger, but now we needed a really efficient and reliable way to detect middens across our search spaces. So we went begging to our collaborators in the computer science department and asked them to please help us develop a computer vision model that could do just that. And while I don't really have time to go into detail in 5 minutes about our AI methodology, I do want to point out a few key challenges that we brought to Liz Bondi and Nikhil Bahari, the computer scientists that help us develop this model. And the first was that we had really imbalanced classes. So if you think about a landscape, there are obviously going to be far more locations that do not have middens than those that do. We also had a really small data set size of only about 80 labeled middens to start. So we needed a model that could deal with these limited labeling resources and also that could consider our RGB and thermal image features. So we were able to adapt an active learning model that incorporated this novel query approach that was able to address some of these limitations that otherwise would have been huge roadblocks to an AI methodology. And when we wanted to test our novel method, we decided to compare it to 2 commonly used computer vision models. The first being a passive learning model that you feed 100% of your labeled data, but requires a really big manual labeling effort up front. And the second was an active learning model that does use intelligent querying, but is usually pretty unstable for small and imbalanced data sets like our own. And we compared these to our novel approach that used just 8 middens as our initial training set, but with this novel querying method, we thought that we'd be able to address some of these limitations. So our preliminary results show that with this passive learning model, again, feeding 100% of our data up front to this simple classifier, It performed pretty well with an average of 75 to 85% accuracy. But again, this required a lot of manual labeling up front. So when we switched to that standardized active learning model, we saw that, as expected, our data was just too challenging and it couldn't cope, and the model didn't achieve a better than random performance. But when we incorporated our novel querying methodology, we were able to get a model that's not perfect, but achieves about 75% accuracy. And again, this is starting with just 8 labeled middens. And if you were here for the AI talk yesterday afternoon, you know that often these classifiers require hundreds or thousands of labeled images. So this is not a bad result. So to give you an idea of what this performance looks like in space, our validation set is here on the left, and the yellow middens are those that are classifier detected, and the reds are those they missed. So we still have room for improvement. And to give you an idea of what this looks like across our entire site, on the right, we have all of the middens that were labeled by our AI and human classifiers. So quite a few across this 300-hectare area. And yeah, just to summarize, we were really able to overcome some huge challenges to an AI methodology for detecting middens and achieve some promising preliminary results, but we have plenty of work to do. I'm recruiting the rest of the Davies lab to ground truth some middens in Kruger tomorrow, actually, and we're planning to apply our model to some new sites in different landscapes in the near future as well. So hoping that we'll be able to dig into more ecological focused research questions and hopefully apply this to some conservation applications in the future as well. And thank you very much, and feel free to find me if you have any questions.
Thank you, Catherine. That was the longest 5 minutes in 11 minutes. Have a chat to Catherine over tea. We're not far away from that. Our next one is another 15-minute talk by Marketa, and that is on some back communities in Kruger.
Good morning. In regard to bats, Kruger National Park is perhaps the most investigated area in Africa, and perhaps even an ideal model to resolve dilemma repeatedly discussed in literature. Arbet communities of savanna biome, unique structurally integrated entities, are just a mere and ad hoc assemblage of the forms originally demanding to other habitats. Our study was integrated into highly standardized settings of complex project Mosaic, already referred in a series of former presentations. Diverse environmental variables were monitored at 60 fixed points at 4 regional systems, each represented by 5 triplets of points differing in access to water. All points were controlled twice, at springtime in November and in late summer in February or March. Community of foraging bats was monitored with aid of whole-night acoustic recordings. The records passed through post-hoc sonographic analysis and combined identification procedures. The microphones were placed by stick at height of 4 meters. This attributed, of course, to certain selectivity of our records, because we have no or only scarce data of non-echolocating bats, of foliage gleaners detectable at short distance only, and ground gleaners with weak echolocation. Yet, quite rich and reliable records are available for aerial foragers like mollusids, which are fast flyers exploiting distant open space foraging grounds. Then vespertionids foraging mostly around vegetation and in semi-cluttered habitats, and one emballonourid species, Taphozous mauritianus. Based on sonographic analysis, we distinguished 25 acoustic parataxa, of which 22 were clearly co-identified with real species. Well, briefly to some results. Here is a graphical summary of abundances and species composition of particular samples. That means whole night recordings from individual points. Vespertilionids are indicated by blue color, molluscids by red. In total, we analyzed almost 117,000 of echolocation records. Abundance of particular samples exhibit quite a large variation. Thus, the first question is, naturally, what is behind so large variation? One pattern is clear. A highly significant differences among the points differing in access to water. Against expectancy, there is only minute difference among this category in community structure. In general, scaling of abundance and species richness of particular samples seems to be proportional. Yet, there are several points exhibit extremely high bed abundances. Thus, another question is by which they differ from the other points. Comparison with a set of contextual variables suggests no clear relation. The result was supported also with a series of ANOVA analyses. Indistinct, yet significant positive correlation were found with one of multivariate variables of plant diversity and bird species richness. But higher correlation was found with distance to the nearest camp. Extremely high bed abundances tend to appear in sites close to camps as a source of roosting opportunities otherwise rare in savanna habitat. We could hypothesize that appearance of trees would have similar effect and ask whether the bed abundances are influenced by vegetation cover. Of course, neither this variable shows significant effect. The centroid of both of maximum and minimum bed abundances show the same position as centroid of all points under study. Well, let us look now at other aspect of our record to the variation in species composition and dominant structure. Here is a list of main parataxa arranged by their total abundances and presence at particular points. Again, molluscids are indicated by red color, vespertilionids by blue. Against expectancy, all samples, all points show nearly identical species composition. Thus, the center of community structure is the same. It's different from the birds. But quite higher differences pronounced between season differences between spring and late summer samples. Spring samples are characterized by predominance of molluscids, but late summer samples show a predominance of vespertilionids or equal representation of both the groups. In short, these between season differences seems to be far the most obvious component of variation in Kruger Bed community structure. Spring data come from the end of cold and dry winter time, a critical period undoubtedly associated with decrease of resource capacity. We hypothesize, by analogy with Mediterranean, that vespertionids capable of active hypothermy decrease their foraging activity and inhabit inside their roosts. While molluscids capable of distant foraging increase their spatial dispersal in search of suboptimal distant grounds. In our opinion, this is the most feasible explanation of between season differences we observed. Here is a map of mean abundance and amount of between season abundance changes. It shows clearly mosaic. It shows quite a mosaic pattern, again without significant effect of contextual variables. Well, summing up, we found that abundance of foraging beds is influenced by access to water and vicinity of roosting opportunities. Both is trivial. Non-trivial is that community structure show almost no variation except for the for a higher between season differences. But what tell our results in answer to the main question of our study? First, let us look at the habitat preferences of individual species represented by mean values of vegetation cover of all records of particular species. Surprisingly, for vespertionids and molluscids, and either in spring and late summer samples, they show almost identical position as common centroid, which is the same as centroid of the points of all points under study. To quantify a structural structural complexity of studied assemblages, we computed a degree of nestedness as a measure of spatial faunal integrity. Here we express the result in a form nestedness matrix and two quantitative characteristics: the degree of ordinary arrangement, which is from 0 to 100%, and mean thermodynamic temperature, which is where 0 refer to a completely rigid state, and 100 to a completely random arrangement. The values computed for our data set show a greatly pronounced nestedness pattern. Thus we can supplement our results with conclusion that habitat preferences of particular community members are in perfect balance to the conditions available in Kruger National Park Savanna biome. And as a high degree of nestedness suggests, the core of bed community became assembled in coevolution with pattern of local savanna biome. All that suggests that bed community under study can be looked upon as an example of highly integrated entity structurally adapted to the conditions the savanna biome does provide. Thank you for your attention and your time.
Thank you, Makita. There's time for one question. All right, thanks, Makita. I'm sure you can talk to everyone. I have some other ideas that you may think about over the next few minutes. We have got two more talks to go for this session. The next is by Rhys Thornley on factors explaining megafauna. Thanks, Rhys.
Hello, everybody. I will try and talk as clearly as possible because I have this really broad Northern English accent that makes it hard to understand anything I say. So, so basically, I'm going to talk to you about the first chapter of my PhD, really where I've been trying to take a step back from site-specific studies and look at the macro-ecological trends in Africa and what determines megafauna biomass in African savanna protected areas. So, really, we need to start by thinking about what we define as megafauna, because that's really not a word that's used very much in savanna ecology at the moment. And so, for my research, what we've done is we've considered anything bigger than a steenbok as megafauna. So the cutoff point was 10 kilograms, all the way up to savanna elephants, and only focusing on large herbivores for my research at the moment, just because the large carnivore data was much more tricky to get hold of. What we need to do is then also take a step back and think about the world that we're in at the moment. So unfortunately now we're in a highly human-dominated environment the world over, and Africa has been no exception. And despite the global cultural value and ecological value of megafauna, unfortunately they have not been exempt from the pervasive human influence that is now pervading the world over. And unfortunately now, a lot of African megafauna are now restricted to protected areas. So protected areas in Africa really now have the key mandate of conserving and protecting megafauna for posterity. But how are they performing to deliver this key mandate? So to understand this, we can go to the literature and look at this study by Craggy et al. 2010. And what you can really see, if you look at the y-axis, that is a 1970 baseline on number 1, and anything below number 1 represents population declines. And what you can really start to see from this between 1970 and 2005, so really since we've been in the thick of the Anthropocene, you can see that for East Africa and for Western Africa and for all of Africa in general, they've seen quite big, substantial declines in the megafauna populations within protected areas, indicating that in these instances that the protected areas are failing to fulfill their key mandate to protect and to conserve megafauna. And we can take another step back and look to the literature, and from the literature we know that certain things drive these declines. And that can be things like poaching, land use change, and systemic underfunding of protected areas, which is common across much of Africa and probably going to get worse because of COVID. But you also need to consider these declines within the context of the people that live around protected areas, which are often situated in some of the world's poorest countries, and some of the most impoverished people also live around them, meaning that often they suffer from severe income and food insecurity, and so have to rely on the illegal exploitation of protected areas for some kind of financial and social security. And so, if we consider the macroecological trends, one thing that really stands out over the Anthropocene is Southern Africa. And so, as you know from the wonderful place that we're in, many protected areas in Southern Africa have maintained and retained significant megafauna populations despite this overall cross-continental decline within protected areas. And so really my key question was, what drives this disparity? What explains this difference? And can we learn things that can improve conservation practice and conservation policy by understanding these cross-continental macroecological trends? And so, thinking about this, I'm really thinking, can Southern Africa be a lesson for the rest of Africa to really reverse this overall decline? I then had this question. So I wanted to know, how do anthropogenic and natural variables explain megafauna biomass across Africa, specifically looking at savanna protected areas? with the assumption that if these declines and these trends are driven by natural variables, you would expect natural variables to be the predominant driver. And if human variables are now having a pervading effect, what you would expect is maybe this to slip away and then for human factors to become more dominant within protected areas. So, yeah, so to answer this question, I worked with some absolutely wonderful organisations, including South Africa National Parks, which was really nice. And they collected and shared their aerial survey data. And what we've compiled from these organisations is an extensive cross-continental overview of megafauna populations within protected areas. And that enabled us to estimate megafauna biomass for 2015. So what we're looking at is the spatial variation, not the temporal variation. So this is basically a snapshot analysis to see what drives megafauna biomass. And what this looks like is 53 species, 43 protected areas, and 550,000 square kilometres of savanna. So it's a huge study, huge scale, going from southern Chad all the way to Addo Elephant Park. So it's quite a big scale, and you know, there's quite a lot of socio-ecological gradients there as well. And then what this enabled us to do was estimate megafauna biomass. And what you can see is you start to get the feeling that a lot of the biomass within protected areas is substantially lower than what you would expect. And so you start to really then think that maybe natural variables aren't actually driving these processes to the extent that they should be. And maybe something else is going on, with many protected areas having significantly lower megafauna biomass and really high hyperconcentrations of megafauna in southern Africa, sort of around western Zimbabwe and northern Botswana, and also in places like the Kruger where we are right now. And so what you can see when you plot these country-wise is that you start to see maybe there is some geographic component, but there seems to be much more variation than what we would expect to be explained by geography just by itself. So what we then did was go to the drawing board and look at what we think drives megafauna biomass, but then you end up with this hideously complicated model that we think there may be national anthropogenic drivers, local anthropogenic drivers, and then local natural drivers that all could play a role in determining megafauna biomass in protected areas. And so to answer our question, we used boosted regression trees, which was a machine learning technique to try and predict megafauna biomass in protected areas from the proxy variables that we have used. And what we get from this is actually quite a good model performance. So 0.74 R squared and a root mean squared error of 3,000 predictions of megafauna biomass, the more important the variable is in driving and determining megafauna biomass within protected areas. And what you find, if you were to sum the red and the green variables, so the red being the human variables and the green being the natural variables, you find now megafauna biomass is determined roughly 50-50%. So we're really starting to slip into a world where humans are having a really increasingly pervasive impact within protected areas, despite the fact that they're supposed to be the areas where megafauna are most protected and most sheltered from anthropogenic disturbance. And of these predictive variables, we find quite interestingly that the Human Development Index, which I'll talk about in a second, and the percentage of open natural savanna have the greatest influence. And so, unsurprisingly, what we see is the model performs quite well because it predicts things that we already know are important for megafauna, such as the percentage of unmodified open natural savanna are important drivers, and also the percentage of permanent water, because we know many megafauna functional types have a high dependence on permanent water, especially in things like the dry season. So we think that the model performs quite well because of these two things, so since income. And what you see on the y-axis, we use partial dependence plots. So this predicts the changes in megafauna by really low. The low level of human development really stifles a protected area's performance for megafauna. Potentially because that could lead to things like a high reliance on the illegal exploitation of megafauna surrounding protected areas in these instances. And then sort of when you get to the lower end of the higher end of the scale, you see that megafauna are released from some kind of human pressure that is present elsewhere, and that facilitates huge increases, really huge increases in megafauna biomass in those instances. And so the second most relevant human predictor was livestock biomass. That seems, from what we've heard this week, that seems to make sense, right? But if you look at the y-axis, what you can see actually is that it causes a really sharp decline, but not a complete decline, indicating that potentially different megafauna functional types are being displaced. So that can be things with similar ecological requirements to livestock, sort of like water-dependent grazers or small non-social browsers that are all being displaced, and then really only leaving behind megafauna that are resilient to these kinds of disturbances driven by livestock. And then also what we find is the Biodiversity and Habitat Index. The Biodiversity and Habitat Index is a measure of a country's environmental governance, but specifically pertaining to protected area management. And a similar situation to the Human Development Index, these real hyperabundances of megafauna, and how does that tie into the Human Development Index? Well, it's a really complex picture, right? We don't have one answer, and there is no one answer. But when you think about Botswana, particularly northern Botswana, what you find is often this, I'm happy to talk about that, it's a very complex picture, right? There's not going to be one straight answer, but when we think about equitable conservation in the Anthropocene, we really should be putting the community at the heart of conservation. And yeah, so with that, I would just like to make the point as well that a lot of people here focus on single study systems or single species, but I think we all have a real big responsibility to make sure that our impact transcends not just a species or one protected area, but is really effective for all of Africa, so that Africa can really make the most of its amazing animals. So with that, I would like to say thank you very much, and thank you to all the wonderful people involved. If you want to give me online abuse, you can follow me on Twitter. Thanks very much.
Thank you, Rhys. We've got time for at least one question. Sally?
Be nice.
Yeah, thanks for that thought-provoking talk. I have 3 questions, but I'm only going to ask
3?
one.
Okay, wow.
I'm going to pick the one that...
Pick the easiest one, please. So.
Well, maybe I just didn't see it. Maybe it's a matter of clarity. I didn't see an index of whether the reserves were fenced or not. And so I'm wondering whether you thought about that.
Yeah,
and
no,
I
definitely.
think it would probably correlate with your development index. So I'm worried that what you're actually showing is whether the reserves were fenced, not an economic response.
Yeah, yeah. No, definitely we considered that, but I think the problem is that we couldn't always get, we couldn't necessarily represent fencing in a reliable way for a lot of the protected areas. So some of the more developed protected areas like here, you can get fencing data, but for some of the more underdeveloped regions, sort of in the Chad or in the Democratic Republic of Congo, then, it's really not available. So it wasn't really, we couldn't reliably include it in the model, basically, because we didn't have access to reliable fencing data in terms of like remotely sensed fencing data or something like that. So we did think about it, but we decided in the end we couldn't include it.
Sally, respect the chair here. We've got one more question here. Sally can ask you the additional ones in tea time.
Thanks, Rhys. That was a nice presentation. I also have a question about a variable that I thought might be important, and I don't think I saw it, which is information on soil nutrient levels, because the idea is that higher soil nutrients support more herbivores.
So we have two different measures of vegetation productivity, but we've not included, but we didn't include soil productivity, because we thought that it would be better represent food availability through the EVI. But I can see what you mean about the vegetation quality, and it's something that we can look at for the before we submit it. So I'm happy to talk about that more later.
Thank you, Rhys. I think those are really important questions that and Sally is going to raise some more later with you. One of the challenges is how do you put soft values in? Like, for example, certainly in Southern Africa, a lot of people only talking about a species is worth it. If it pays, it stays. So think about how to put those sort of things. Yeah. Thanks for that, colleagues. And sorry, Sally. All right, we're in our last presentation for the morning. We're going to go to the fire side of the world again. And Genia will talk to you a little bit about immediate and long-term effects of fire on savanna systems. Thanks, Genia.
Hello, everyone. Today I'll be presenting about some of our results from effects of fixed-term fire regimes on savanna vegetation structure and above-ground biomass. And in the end, I'll also be showing some glimpses of our ongoing work about variable fire regimes. So we all agree that fires are an important component of savanna functioning, not only because they exert strong top-down controls, but they promote structural heterogeneity. Two important aspects of fire regimes are frequency of occurrence and the intensity of burn. And both these factors create different levels of heterogeneity. For instance, fires of high frequency can cause the woody vegetation structure to remain under a certain height by creating a mechanism called as the fire trap, where all the woody vegetation are constrained to a lower woody resprouts. Similarly, fires of different intensities can create different levels of heterogeneity. For instance, a hot, dry season fire can curb the shrub encroachment, but at the same time can lead to the loss of large trees. So by keeping all this in mind, like what we are interested in, we are interested in knowing how the variation in fire frequency and intensity affects the savanna vegetation structure across landscapes, varying in mean annual precipitation. But we already know that fires exert strong control over the vegetation, which is in the high rainfall zone, than as compared to the vegetation, which is in the low rainfall zone. But the quantitative measure of this variation across the mean annual rainfall gradient is missing. It's largely because of the absence of the fine-scale vegetation structure data. So in this study, we seek to answer, like, how does the variation in fire frequency and intensity affects the savanna vegetation structure across landscapes that vary in the mean annual precipitation? And we looked at 3 vegetation metrics, which are above ground biomass, tree density, and vertical height profiles. Experimental burn plots in Kruger National Park, and these experimental burn plots are located in 4 different landscapes of Kruger, which are Mopani, Satoras, Cuckoos, and Bitterscope, and all these experimental burn plots have different rainfalls. So we looked at all the fire frequencies, that is the annual, biennial, triennial, and for Mopani and Satara, we even looked at burning every 4 year and burning every 6 year. For the fire intensities, we took fire season as a proxy. To obtain the fine-scale vegetation structure data, we used the UAV LiDAR remote sensing technology that gave us high-resolution 3D point cloud data. And our UAV LiDAR data acquisition was done during the mid-wet dry season from January to February 2020. And the final point density of our LiDAR point cloud was 150 points per meter square. Here is a side view of the 3D data of one of the experimental burn plot in the wettest site, that was Bitterscope. And here blue resembles ground, and the red points resemble more taller vegetation. And here's some more LiDAR transits across different burn treatments during early dry season burning in one of the wettest site, again in Bitterscope, and I think this is from the Shabeni experimental burn plot. And we clearly see like how these different burning regimes or like different fire frequencies imparted different structural heterogeneity. So to obtain the above ground biomass and tree density, we used the canopy height model at 10 centimeters resolution derived from our LiDAR data. And you see it on right, with the height going from 0 to 12, these lighter shades depicting the ground, and the higher vegetation depicted in the green color. So we did a dynamic moving window treetop detection, and then that canopy height model was segmented with the help of the watershed segmentation. This segmentation gave us the crown area and as well as the height estimate of every individual that was there in the plot. And these measures of canopy cover and as well as the height were fed into a biomass allometric model, which gave us the biomass for every vegetation individual in the plot. And since we did the segmentation, so we got the estimate of the tree densities per plot as well. So let's start to look at some of the results. I know like there are like a lot of these box plots, so we will try not to be like overwhelmed by these, and we'll go like one by one. So on x-axis, we have the landscapes in the decreasing order of the mean annual precipitation, going from Britoyoscope to Mopani, so that's the decreasing rainfall. And on x-axis, on y-axis, we have the above ground biomass. And every figure here represents different burning season, where December, February, and April represents the wet season, and August and October represents the mid-dry and late dry season. So the general conclusion is that the largest biomass reduction occurred in the dry season burning, which has been already there in a lot of the previous researches, which were looking at the woody canopy cover, like how the woody canopy cover is varying across the seasons. But interestingly, we found 3 key observations from these above ground biomass results. First one being, if you look at the wet sites, which are the Britoyoscope and Skukuza, in the wet season burning, we found that the highest or the largest biomass reduction occurred by the lower fire frequencies than as compared to the higher fire frequencies. Again, in the same wet landscapes, that is Britoyoscope and Skukuza, we found that during the dry season burning, it was the higher fire frequencies that caused largest reduction in the biomass than as compared to the lower fire frequencies. Another interesting finding was also that if you look at the most driest landscape, that is the Mopani, except of burning every 3 years in the February season, every fire frequency, irrespective of the season, caused significant change in the biomass, kind of pointing to the direction that for landscapes like Mopani, which are drier, it's the presence of fire that constrains the biomass, not the fire frequencies. And we also looked at the tree densities, and we observed that there were alterations in tree densities across the burn treatments. One of the interesting results here was that if we look at the driest landscape, which is Mopani, we observed that all the burn treatments had more trees than as compared to the control plots. In contrast, we observed a declining trend in tree densities for the wet landscapes, and that was mostly during the dry season burning. Another interesting thing here is that although both Britoyoscope and Skukuza have just a difference of 100 millimeters of rainfall, the proportional decrease in the tree density was more at the Skukuza site than as compared to the most wettest site, which is the Britoyoscope. One more interesting observation that we found was that in some of the wettest site, in the burning seasons, we found an increase in the tree densities, as you see here in the late dry season burning, in the most wettest site during burning every 2 years, that there was a significant increase in the tree density, and again here as well in the triennial burning. Actually, these increases were happening in the shorter height classes, and we will see them later in the vertical height profiles, that where exactly these increases are happening. So we were interested in knowing what are the important factors that are constraining the above ground biomass and tree densities. So for above ground biomass, we found that the most important model was which contained the terms landscape plus season, while for tree density, we found that it was only landscape that constrained the tree densities. But the second most important model for tree density was also landscape plus frequency. Then we looked at the variable importance, like what are the important variables that are constraining the above ground biomass and tree density. And we found out that it's the fire season that is influential in constraining the above ground biomass, and while for tree density, it was majorly landscape. So we also looked at 3D vertical height profiles. So on x-axis, you see here the LiDAR return frequency, and on y-axis is the vegetation height. So this LiDAR return frequency is what they were derived by voxelizing the point cloud. By voxelization, I mean like simply like you just put or spread like numerous number of cubes all across your point cloud. So we chose a voxel length of 0.5 by 0.5 meter. So now you have your point cloud, you have all your voxels, and for every voxel, you know at what height they are, and you know like how many LiDAR returns are passing through those voxels. And that's how these LiDAR return frequencies were created. We found some interesting trends here that fires imparted unique vertical niche spaces, and for Proteuscope, these unique vertical niche spaces were in the canopy height of 3 to 7 meters, while for Scucusa, they were somewhere around 0 to 2 meters. And when I was talking about in my previous slide that there was an increase in tree densities in some of the burning treatments, that was mostly occurring in the shorter height class. So you see here in the October and as well as in the April. So for the drier landscapes, for instance, the Satara one, which has high herbivory density, we found no significant differences between the treatments and the control. The only significant differences between the controls and the burn plots were during the dry season burning, but there was no significant differences between the treatments. And similar thing was there in the Mopani as well, that there was a significant difference between the control and the treatments, but there was no significant difference between the treatments. Again, pointing to the same thing which I mentioned earlier, that from our data, it looks like that for dry landscapes, it's just the presence of fire that constrains the vegetation structure. So to conclude, so these almost 70 years of fixed-term burning exerted strong top-down control on the vegetation, and these structural changes imparted by fires varied across the landscapes, meaning the effects of fires are context-dependent, and also highlighting the relative stability of savannas. We also found that fire frequency and season operated contrastingly on the above-ground biomass and tree densities, where fire season was more influential for above-ground biomass, while for the tree density, it was majorly landscape or fire frequency. And we also saw that fires created unique vertical vegetation niche spaces. But what are the management implications for these results? Like what do we kind of like give suggestion? Like what suggestions can we give from our results? So I have put again the vertical height profiles of the Proteuscope landscape. On the x-axis is the LiDAR returns, and on y-axis is the canopy height. And in the August vertical height profile, these gray color lines are burning annually, and then yellow ones are biennially, and the blue ones are triennial, and the green ones are the controls. And we clearly see here that in the August, which is like mid-dry season burning, that this burning regime, annual burning regime, gets rid of the shrub or the shrubby vegetation, but at the same time retains the large trees. That's not the case in the late dry season burning. We get rid of the shrubs, but at the same time, we lose a lot of the large trees. So if the goal of the management is to prevent the loss of large trees, our data suggests to go for the mid-dry season burning than as compared to the late dry season burning. But at the same time, I'll also like to kind of state this thing that our data only looked at the vegetation aspect. We didn't consider any other aspect. We didn't look at the below ground carbon pools, or we didn't look at any other biodiversity or heterogeneity aspect. So this is like something that comes only about the vegetation structure data. So all these results which I presented right now, they are from the fixed term fires, where we considered fire season as a proxy for intensity. But in order to understand fires, like how they constrain the vegetation structure, we need to look into more variable fire regimes. And also, we need to start to begin to account for vegetation change between before and after fire events. So in order to do that, we started looking at the fire experiment set up at the Lower Sabi region. And we thank all the collaborators, especially Sally Archibald, Carla Staver, Kate Parr, and Sen Parks, for setting up this experiment and giving us an opportunity to look into these fires. So in this experiment, they have set up almost near about 31 hectare plots, where like each of the plot burns during different time of the day and having like variable fire regimes. So we collected LiDAR data for before and after fire for every plot. And on the left, you see before fire, and on right, you see after fire, the patches created by the fires and the fire square. So we compiled all the data for these plots, which were burning at different times of the day and had different fire intensities. And we looked at the density estimates of the canopy height. So on top, you will see the time of the plot, like at what time it was burned. And now I'll just play a GIF, and we can see like how the mean of before and after canopy height changed. So you'll mostly see that fires during morning were much more cooler, and the mean of the canopy heights were nearly like, it didn't drift away quite a lot. But as you go more towards the afternoon or noon fires, the mean shifts more towards 0. So that means like you are losing a lot of the vegetation. So I'll play it again so that people see it again, that the morning fires were much more cooler, and the afternoon fires, especially around 1454, there was a huge drift between the canopy height between the before and after. So we not only are looking at the changes in the vegetation before and after fire, but we also collected thermal data during the active fire for every plot. And the thermal data was collected every 2 seconds, which you can see here on the left-hand side, how fire progresses into the plot. And we are working on creating some of the fire properties, such as the residence time and intensity, and as well as rate of spread, and linking it. I'm almost done.
Please finish.
And on right, you see the vegetation structure change. So that is like something our ongoing work, which we are doing and hoping to come out with some interesting results. And with this, I conclude my presentation, and thanks to Sandparks for all your help during the data acquisition. And I also thank Milton Fund from Harvard University that helped us to collect data for our projects. Thanks, everybody.
Thank you, Gina. You can have a further discussion with Gina in the tea time. Thank you to everybody for participating in this session. It was a good, varied session. We managed to deal with a power break very well. And yes, apparently, one of our attendees, it's his birthday today. Lawrence, are you in the room? Lawrence Swanepoel. Well, if he's not, if you do see him along the line, give him a hug or something like that, you know. But remember social distance or something. But thank you, and enjoy tea time. I think we're coming back at 11:15. Thanks.
Morning. I'm wondering how, is that okay? Yeah, it's okay, I guess. Yes. In front of my face? Okay, okay. 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 Thank you very much.
Thanks, everyone. So we're ready to start the next session, which is around land surface patterns and remote sensing tools. So Sam did a great job this morning before tea, keeping people to their time. So I'm just going to follow in his footsteps. So our first speaker is Christiana, and she's got a 15-minute talk, and she's going to be telling us about their Coldems activity. Thank you.
Okay, thank you very much, Judith. It's a pleasure to be here in person. It's just wonderful to see real people again, three-dimensional, feel them, etc. But we all know that. So, this is a very long title, but what is important is this term Coldems. That is our SunParks project, and it is combining different projects on this bilateral cooperation between South Africa and Germany, the Spaces program. That's why you have the ministry and FONA down there. And the two projects that I'm representing is SALDI and also EMS Africa. Okay. So, I left out all the organizations before, not to confuse you. I have many slides and overwhelming information, I'm afraid. But so we are very happy in a very large cooperation context with several South African organizations and institutions, and the same from the German side. And we have also the pleasure to work in Kruger Park at least for about 15 years. But with the SALDI project, we have been extending our sites. So we are looking at 6 100 by 100 kilometer sites across a climate gradient. But it's not only a climate gradient, obviously this influences the land cover as well. So these 6 sites are very diverse. And I'm going to give you really briefly and shortly results of 3 of the sites. And in all of these sites we have protected areas, national parks, and obviously areas under human use. So for those of you, not everybody is a remote sensing geek like I am, since I can think, I guess. So just a very brief introduction on what the data is that we are using. So if you're looking at optical data, and maybe many of you are familiar, of course, with air photos or with optical Landsat data, since 2017, we have a new sensor in space run by the European Space Agency and the European Commission. It's the Sentinel fleet. It's a new era in remote sensing because these satellites have a better spectral resolution, more channels that cover the Earth, and also a higher temporal repetition because there are always at least 2 satellites, A and B, in space. Now C is being launched, and this program has been guaranteed for the next decades. Well, hoping everything stays peaceful, I assume. So it's also 10-meter resolution, so we have higher spectral resolution, higher geometric, and higher temporal resolution. So what does optical sensors see? Obviously, the optical and the near and middle infrared electromagnetic ranges. And so what we measure in an optical satellite is the reflectance of the sun depending on the state of the surface. What we are using in our projects are also the radar satellites that cover the range starting about 1 millimeter, 1 centimeter, about that range, but I get to that later. So these satellites are called Sentinel-1, and again, there's A and B, and for South Africa, this meant that we have every 6 days, we have an image, and since radar penetrates clouds, we have a guaranteed image every 6 days, so wonderful time series. And here's an image of that marvelous Sentinel-1. So they are identical, Sentinel-1 AB. Now, I would assume that not all of you, but maybe I could just ask to get you in active modus. Who knows anything about radar remote sensing? Please raise your arms so I know I'm spending only 1 person. Oh, 3 people, 4. Okay, so please pay some attention to this slide because that is the basis of the further analysis. Well, radar is using very long wavelengths, and we have some satellites up in space and coming up with long wavelengths, 23 centimeters, almost like a page length. And they, of course, here indicated by the yellow arrows, they penetrate vegetation much more. You have returns from the ground. So they are famous, these sensors, for mapping soil moisture or even flooded forests, flooded tropical forests. But the workhorse that we are using, Sentinel-1, A and B, has this wavelength, 5.5 centimeter. This wavelength was decided by oceanographers about 30 years ago. And we stuck with a wavelength that is very, very, let's say, unfortunate for land surfaces, because you see here it's stuck somewhere in the vegetation. Yeah, it may penetrate if the vegetation is not very dense to the ground, so you get an estimate of the surface moisture, but only if the vegetation is not very dense. So what I have to mention here is now about these. One thing is, what is the radar wave interacting with? It is with water molecules, like your microwave in your kitchen at home. You put your chicken in and the water molecules start rotating, or your coffee, and it makes it warm. So we see water either in the vegetation or on the ground. Now we are also using, this is a further term I will be using, coherence. Coherence is a so-called interferometric product. What it is a correlation between two radar images. Now, looking at this correlation, the degree of correlation, it tells you something of how this vegetation volume is structured. So it actually tells you more about the geometry of the surface, and especially of the vegetation. So this is where C-band now becomes interesting, because we know it's stuck somewhere. The 5 centimeter interacts with 5 centimeter things on the surface, so leaves, for example. Okay, our SALDI workflow, but we are also here partnering obviously with the EMS Africa project, is actually oriented, and SALDI stands for land degradation, South Africa of course, and in index development, is oriented towards 4 assessment procedures that Graham von Maltitz published 2019. So we want to contribute to rangeland degradation, bush encroachment, degradation of croplands, and he also, of course, focuses on alien species, but we cannot see that from that type of sensors we are using. So we're looking at vegetation cover change. So and these are the products that we can produce from remote sensing. Because in the end, I mean, this is the wish list, but this is what we can produce. Now, looking at our inputs, Sentinel-1, we have the backscatter, of course. We have that coherence map that I just explained. We have various ratios, and we have multi-temporal statistics. And that's the power of radar, that you have nice time series. Of course, you have time series on the optical products as well. You have a whole range of products that you can produce operationally. Software is available, free in the internet. So the derived products here is breakpoints. Some of you may know this from also optical time series. So looking at changes in these time series, you can do the same with the radar satellite. And we are producing woody cover, and now it gets interesting. A woody cover product from radar, a woody cover product from optical Sentinel-2 data. And here, of course, with radar, we try to produce a surface moisture product. So this is where we want to get it, synergistic products, exploring all the advantages from optical and radar to come up with these 3 types of products that eventually lead to a land degradation monitor. Now again, I will start here with Eshlanseni site, which is covering southern part of Kruger National Park. I will then show some results from Mokala National Park and Ben Fontaine Nature Reserve, and we then finally move to Ohabe. So very quickly, I just move through the results, and you have the slides anyways, and get in touch with us if you have further questions. Okay, this Cold M's Sun Parks project enabled us to produce some first-time products. So one is the just published in December 21, the first sub-meter resolution digital elevation model with a 25 centimeter pixel spacing. So I just show this slide to point out the great support we have here from South Africa with something screwed up here with the format of my slides, but anyways, Geospatial International, you may know, and with a wonderful contact with Wolfgang Lück, formerly with the PCI Geomatica company, now Catalyst, we had the possibilities to process this huge amount of data. So this is a reference that is accessible here through an archive. What you get is a wall-to-wall surface terrain model, a wall-to-wall surface model, and a wall-to-wall canopy height model. And you can already see with 25 centimeter pixel spacing all the great detail. So this is like a reference for us also to develop the woody cover maps, all the further retrievals that we need to do, because this will also help us with a very high resolution to validate, for example, the soil moisture maps, where is vegetation, how much vegetation is there. So result number 2 is, and please, if you go to the poster, I don't know why here the format screwed up, I'm sorry. So this is an analysis of our four terrestrial laser surveys at the Skukuza Flux Tower. Where we want to get it is to measure again, the biomass to a very high accuracy, to use it as a validation tool for our woody cover maps, but also to see what is the limits of applying terrestrial laser scans to multi-seasonal scans. So these scans start in 2015 and they cover 4 extreme seasonal conditions. So this is the basis for our validation. Here a first was published in 2020, first 10-meter pixel spacing woody cover map wall-to-wall of Kruger National Park. And what is interesting for us as remote sensing, as mappers, that of course you're reducing the resolution. This is a known fact, but it is important if you use this product in further modeling, what are the accuracies, and you see the more you generalize, the better the accuracies get. So this was trained only with a small LiDAR, airborne LiDAR strip, but now we are moving on to including JEDI, of course, and we are working with Xenia together, to include more accuracy measurements. Now, this is also a woody cover map from Sentinel-2. There is a poster out there, and what, of course, we want to get it is mapping change from these products. But to map change, you have to know the accuracy of each individual product. That's why we have that focus on the high resolution. Also in progress, surface moisture, because here you have to exclude, of course, where you have vegetation coverage. That's why it's so important to get these synergistic products. And here you can already see this was after the large drought at the end of the dry season, but you see the annual differences of the surface moisture content. So again, a valuable environmental product. Okay, I move over this because you can look it up in the slides if you're interested in these kind of optical products. For those of you fans of image processing, Tesla cap, I'm a big fan of this, but now we're looking at 20 years of Tesla cap explorations, starting with the Landsat on 30 meters and now having a product that includes Sentinel-2. And this is where we are now working on. This is Benfontein. We are looking at single pixels. And what we will see here is the mean, median, and standard deviation of 100% grassland. This is from, also covered here now, from Nangula Ngialva. She has also a poster out there. Please have a look if you are interested. And what we try to do here is a sort of pixel unmixing, but related to radar. Now you're looking here at the Mokala National Park region, and what we did is using the coherence time series, again at what I have explained before, to develop a grassland community map, to start with that based on these pure pixels, and then identify the radar and coherence returns from the bush pixels and tree pixels to come up with a multi-temporal model to be able to monitor what is, for example, how are the large trees growing or disappearing? Is there bush encroachment or not? How are the plant communities developing? And I've finished with Ochabis. Many of you probably know that wonderful area and all the irrigated fields along the Orange River. But what we are interested in is how is the surrounding surface developing. So one very obvious product, and again, we are using the radar coherence here, is the increase of the irrigated areas, but that's something you can do with optical also nicely. But what we are looking at and interested in is the erosion features in the gullies. So, and again, here radar comes in nicely, and I finish now with my last slide. So this is a visual summary of also products I have not discussed here in detail because of the time, but all of this goes into an open data cube, the SALDI data cube that everybody can access and use with different procedures for the analysis. This is here near Douglas, from radar to optical, just to illustrate what I have been talking about. Now, of course, the big question, what's next? And what is next is continuing the accuracy assessments. So this is where we strongly need interaction with you in all 6 sites. Please contact us if you are interested to join. Then further synergy explorations, and time is out. Of course, change measures, but last but not least, also a connection between in situ point sites and our time series from satellites. And most probably there are some deep learning mechanisms should be employed, because you see the overwhelming data amount that we have. Last but not least, many thanks to Sunpark staff, to all the section rangers and game guards that supported us, and of course to the projects that allow us to do this. And with this, my motto is treat each pixel with respect. It's a unique geometric, spectral, and temporal measurement. And thanks for your attention. I have to run to give this.
Thanks, Christiana. So that was pretty close, but you just kind of a little bit over, but you know, I'll give you that one. So now we're still staying with Coldems, and we're going to move on to Juicy, and he's going to give us a 5-minute talk. Thanks, Juicy.
Yes, hello everybody. I'm speaking basically about the same project, about this Coldems, and I will actually provide the solution of the acronym which we found here, which probably is influenced by the fact that usually before we come here down to Skokusa for the Savannah Science Network meeting, we spent a few days skiing in Finland. So Coldems stands actually for the contribution of these 2 spaces projects, SALDI, South African Land Degradation Monitor, and on the other hand, the EMS Africa, the Ecosystem Management Support for Climate Change in Southern Africa, both funded by the German ministry. The general aim of Coldems is actually to monitor and model land surface dynamics in areas with currently low human impact, pristine areas across a climate gradient in South Africa. And you've seen this figure here already in the talk by Chris. These are highlighted the areas where we have, or used to have, sandparks areas included in Sulplati, close to Kimberley. We were active in Mokala National Park with a soil moisture measurement site, but that was unfortunately destroyed, and so we had to change that to Benfontein Reserve. Our recent ground truthing activities include a lot of surveys, basically gully erosion surveys, using GNSS systems, UAVs by a South African company, because we are not by ourselves allowed to fly in national parks, and terrestrial laser scanning here, for example, at Kruger National Park. The other ground-based activity is basically maintenance of soil moisture sites, which had some kind of surprising fates, actually. If we come to some results, this is actually shown as well on the poster outside. Yeah, it's the survey of a small gully system close to the Skokusa flux tower at the S65 survey since October 2015, so by now over 6 years. And over the 6 years, the main gully had retreated by closely 4 meters, so we have something like 0.75 meters mean retreat retreat of this gully head due to runoff from the S65, which is close by, and it will take another 30 meters until this gully will then finally hit the road and start to erode probably the road there. Gully erosion is an issue which we are looking at, and we were looking at some gully erosion protection or stabilizing features in Mokala National Park. You can see here an aerial photo. What you can't really identify is fascines and sediment fences which were built up here to prevent surface runoff, provoking gully head retreat, and on the other hand, an action which was tested out there, putting up micro ponds which should increase the water retention on the soil surfaces and prevent runoff generation. I told you about our soil moisture measurements. This is the example from Ograbis Falls National Park. A very nice time series here from the beginning of 2019 until January 21, when a big rainfall event caused the logger to be drowned and to be unfortunately taken out of working. To have some more results or look at some more results, I just want to highlight that we have a number of posters still hanging out there where you could get an idea about other activities, other results. And with that, I would like to thank you for your attention and the support of Sandpark's staff, scientific services, section rangers, and everyone else. Thank you.
Thanks, Jussi. I see your time is up, but we will have these slides available on our website as well. So if you'd like any more detail, please just contact either of them. So we are still staying in this session, and we're focusing on soil erosion again. And our next speaker is Lazarus, also with a 5-minute talk. Thank you.
Thank you very much. Mine is a speed talk. I'm going to talk about soil erosion in the Savanna biome, focusing on South African national parks. Why the Savanna biome? This biome is very important because it is home to quite a number of large mammals, which actually play a very important role in the tourism sector, knowing that the tourism sector plays a very important contribution to the economy of South Africa. So we need to take care of the soils and also understand the level of erosion that is taking place across national parks. So repetitive inventories are a necessity, as they play a very important role in promoting management dynamism and pragmatism in dealing with environmental problems. So in this study, we tried to do an inventory through a mix of methods and also a multidimensional approach. So our study focused on the national parks in the Savanna biome, as shown in that map. Of course, Mokala Park is, also belongs to the other biome, which is the Namakaru, but the greatest part of it is also in the Savanna biome. That's why we also included it in this study. So that slide is showing the approach that we used. We didn't want to depend on Earth observation systems only, but we also wanted to integrate social science systems so that we really understand what the end user perceives in terms of soil erosion. So we used GIS remote sensing, basically the SLEMSA model. I will explain about it in a few slides. Then we also used some questionnaire surveys, site observations, interviews with people who use the park, the tourism managers, the park managers. We also try to look at the scientific reports that have been produced by Sandparks, very important scientific reports from which we used data on a vegetation condition index, normalized difference vegetation index, soils data, the geology, to understand what is going on, to be able to explain soil erosion, vulnerability across the parks. So in terms of the data that we used in this research, we used a 90-meter resolution digital elevation model. Of course, we are looking forward to improve in terms of resolution there, because there's now the Copernicus 30-meter resolution DEM. So I think if we use that one, we will also improve our results. Then we also have some soil maps that we use, the Landsat satellite data, Sentinel-2 data, and also rainfall data. So that's the model that we used, the SLEMSA model, the soil loss estimation model for Southern Africa. So this system takes into consideration the physical systems, including vegetation data, climate data, soil data, topography data, right? And from that data now, we determine the control variables which we use to calculate the sub-models, which when combined will give us the main model, which is used to assess soil loss per hectare per year. So in terms of soil erosion hazard classification, we used our quantitative data, and we also used qualitative data, looking at how people perceive it, how they classify soil loss, right? So those, the classifications there are based on the perceptions by tourists. So the surveys that we used, tourist surveys, like I said before, interviews, site observations. Then our results show that soil erosion is not really a big threat across national parks, but you can see that just a few parks are also threatened, like Marakele. As you can see there, we have those classes, the severe, very severe, and also moderate class of soil loss is also found there. That graph is again showing perceptions by tourists, as well as the erosion severity index calculated from our satellite data, and it's showing that really soil erosion is not a big problem, but in Marakele, followed by Mapungubwe, those are some of the parks that are heavily affected. So these are the maps which are showing the distribution in terms of soil erodibility. That is for Marakele, that is for Mapungubwe, that is for Mokala, and for Kruger. Then for Kalakadi. So what are our conclusions? The assessed national parks in the savannah have different levels of vulnerability. Marakele is the most vulnerable. Kalakadi is the least affected by water erosion. Mapungubwe is moderately affected. Kruger Park is not vulnerable, but there are some points that we also observed on our images which show that there is some severe patches of erosion, especially near water holes and abandoned or decommissioned roads. So although in the same biome, parks are exposed to different historical land use practices and biophysical conditions, and consequently management regimes. So we recommend that we implement park-specific control mechanisms that are suited to the historical and biophysical conditions of the park. Thank you very much.
Thank you, Lazarus. Your time is up, so people will catch you at lunch. So now we're moving on to mapping and monitoring vegetation patterns. And our first speaker is Tulia, and she has a 5-minute talk. Thank you.
So good morning. Today I'll be talking about our analyses on historical aerial photography to better understand long-term changes in the vegetation dynamics. So we started with a data set that was acquired in georeference in collaboration with Sam Parks and the Italian Corporation. The available high-resolution imagery dates back to 1940, and the latest is from 2018. But for this study, we only focused on the images from 1940 to 2012 to obtain a map of woody cover vegetation. So there were two main challenges in this study. The first one was to tackle the immense vastness of the Kruger, and the second was how to extract information on vegetation from black and white images. So to tackle the first challenge, we decided to focus on vegetation types. More specifically, 5 vegetation types in the southern part of Kruger. And within these 5 vegetation types, 5 subplots of about 25 kilometers squared each, which is about 2,500 hectares. So to tackle the second challenge, which is to extract information from vegetation from black and white imagery, we decided to use object-based image analysis, which segments an image, grouping pixels together into vector objects. This is called segmentation. And then training and test samples were collected in 2 classes, woody and non-woody cover. And then we use machine learning to classify the images in land cover classes. To perform this object-based image analysis, we used the Google Earth Engine as a tool because it was freely available and it was also easy for different operators to share scripts and images. So now I'll briefly take you through the data set, show you each subsample separately so that you can see all of the data set. So this is subplot 1 of Cobratum Collinum, and as you can see on the left, the images from 1940, and on the right, 2012. And here's the result of the object-based image analysis. This is subplot 2 in the low veld sourbush veld of Praetoriuscope, and this is our classification. This is in the Malelane Mountain bushveld. This was one of the hardest subplots to analyze, mainly due to the mountainous area and because also of the presence of water. This is the subplot 4, which is just south of Skukuza, actually. And this is our classification. And this is subplot 5. We decided to also consider a plot in the basalt area. So this is our classification, mainly to see the difference mainly between the granite and the basalt. So just to take you through our initial results, what we found actually was in subplot 1 and 5, the vegetation actually decreased from 1940 to 2012. This is over the entire subplot. And subplot 2 and 3, the total cover actually remained constant, which was quite interesting and strange at the same time. And then in subplot 4, just south of Skukuza, the vegetation increased. So the next step, obviously, is to scale up our vegetation type. So this is subplot 2. So this is just like 5% of the entire cover of the vegetation type. So this will be particularly hard, as you can see fire scars. This is 2012, but we also have the image from 1940s. And then after this, we would like to obtain a complete land cover map from 1940 to 2012. And hopefully see if this is linked to any past management and elephant population growth that has affected current vegetation dynamics. And then lastly, I just wanted to show you, while we were going through the images of 1940, we came across some settlements, so historical settlements, as you can see in the image on the far right. And we would like to try and use the object-based image analysis to see if we can track historical settlements inside the park. So this is how it is now in 2012. And this is in the area just north of Crocodile Bridge in the basalt area. But we also found also other settlements that we are tracking. So for any further questions, you can ask me now if I haven't run out of time, or this is my email, and this is just to show you Skukuza's camp in 1940. I thought that would be an interesting image for all of you. So thank you.
Thanks, Julia. Your time is up, so we're going to be moving right on. So our next speaker is Mulalu, and we're then going to be looking at some dynamic global vegetation modeling. Thank you.
Good afternoon. Thank you, Chair. Today I'll be presenting a part of my PhD study, which is on net primary production, looking at drivers and trends in Southern Africa. The presentation is divided into 2, so it's the net primary production and also looking into biome distribution. I was hoping that I would be able to be presenting full results with regards to biome. However, I came across some challenges, and I will definitely hope that this will be a good network where we'll get some feedback on how to proceed. So, challenges of mitigation and adaptation of climate change can be highly supported by dynamic vegetation models. That is why we decided to look into Southern Africa, which is defined by the longitude and latitude written there, and for the time period 2002 until 2015. We decided to make some simulations, net primary production simulation using dynamic vegetation model LPJ gas, and compare our results with Earth observation MODIS data with NDVI, since it works as a proxy for NPP, and also NPP. This was to get results on what are the patterns of net primary production in the region and what actually drives those patterns. So looking into spatial distribution of the two models, that LPJ gas and MODIS, we see a great agreement between the two. What you see is the LPJ gas on your left and the MODIS data on your right. This shows a good correlation of 0.7, which was very helpful. So further off, we decided to look into how these two models agree with each other per grid cell. And what you see is, what you see is the Spearman rank correlation per grid cell on your left with the significance on your right. We see that there is high correlation in most parts of the region, and where there is high correlation, we see that there is significance in those high correlation. Looking into the temporal distribution of the model, we see that the two models actually follow the same interannual variation, which was also in accordance to the spatial distribution. So, as I mentioned before, our target was to look into the trends. What are the trends for NPP? So we decided to look into the trends for both MODIS and LPJ gas NPP. What you see is the MODIS NPP in green and LPJ gas in red, and then we also included the proxy NDVI just to see how it looks. And so far, this is across the whole region, and what you see is that an increase in trends for all the models. So this was not really enough to actually seeing that South Africa is more variable and diverse. So we went on to look into the trends per biome. We decided to use the SANB biome map, since it's well researched. And what we found was that the trends we see an increasing trend for all the biomes, except for the desert and also the grassland. However, the trends that we see here, they are not significant except for MODIS NPP in the fine boss and in the forest. We further on looked into drivers of NPP. We did this also per biome. And what you see is that we decided to look into the drivers, looking into climatic parameters. We looked into precipitation, temperature, and solar radiation. What we found was that precipitation was the main driver of NPP in the region for all biomes. And this is collaborated with the Spearman rank correlation, which I did for each model with per biome. What we see is that there is a high correlation for LPJ gas with precipitation than MODIS and NPP and with NDVI. And what we also saw was that there was no correlation with temperature, and this is per biome. And only correlation which was observed was with MODIS, and that was a negative correlation. And we also saw a negative correlation for solar radiation. We also further looked into the impact of carbon dioxide, if this was an influence or a driver of NPP in the region. So we looked into the model for the time period 1900 until 2015, just to see if we fixed the carbon, if that had any influence. And we see that there is a strong increase in NPP in this time period, and this can be attributed to atmospheric CO2, that is CO2 fertilization, and the effect is around 18% for until 2015. So the lesson learned for this study was that we found that there is good spatial and temporal agreement between MODIS and the LPG gas. And this gave us some sort of relief, knowing that there is some confidence in our model. We also saw that precipitation is the main driver of NPP in the region across all biomes. And we saw a generally increasing trend of biome. However, they are not significant. So at the end of the day, we see that the model is good enough for future projections in Southern Africa and may be used accordingly. And that is our future steps. So going forward into the ongoing research that we have, our model is, it doesn't really depict, so it's on biomes. It doesn't really depict the Sunbee distribution, biome distribution as we had hoped, where there is, we find that there is no grassland biome. So the grassland biome is full of trees, and we were trying to figure out how we can make our biome to actually represent the Sunbee biome. So what we did is to hypothesize and say that minimum temperature actually controls grassland. And this was based on the two papers by Wakaling et al. 2012 and Stevens et al. 2018. And these publications were mostly looking at why there are no trees in the grassland. So basically, we thought that the temperature in the region is too low for trees to actually grow to a point where they can actually avoid the tree line. So, however, so for this, we made a histogram just to see between the different biomes. And for the case of this presentation, I'll just look into Namakaru and grassland. So, although the hypothesis, we found that the hypothesis is not really sufficient to support this, is not enough, seeing even if there is some sort of differentiation, it doesn't really stand on its own. So we continued to test this hypothesis by looking at different minimum temperatures. What you see is the minimum temperature at 2, 4, 5, and 6. And we can see that the grassland, the tree line is continuously pushed back. So it doesn't really, it's not constant. So this was a bit of a setback because now we're not sure exactly what other limits we can use. We also looked into water deficit and still looking at desert and Namakaru, we see that at least this is a bit of better representation and maybe it can actually help us. However, we didn't do any simulation with regard to this as yet. But at the end of the day, I need some suggestion on how to proceed forward with my study. Maybe I can get some feedback from you on how to get our model to represent the biome, how to keep the trees and shrubs out of the grassland, and also, yeah, what other limits can we use or input in the model? Does implementing the drought indices, is it gonna work? Or investigating soils? Yeah, thank you. I would like to thank I'd like to thank my collaborators and my sponsors. Thank you for your attention.
Thanks, Malala. We do have 2 minutes, so if anybody has a solution to your problem or would like to ask a question, please go ahead. Okay, no, this is a tough one. Looks like they're also having trouble getting the trees out. Thanks, Malala. I'm sure people will discuss with you more after this. So now we're going to move on to the last presentation from Martin, and it's going to be looking at the seasonal rivers, and it's also a 15-minute talk. Thanks, Martin.
Hello, ladies and gentlemen. My name is Martin Heide, and I would like to briefly report on the results of our project that was aimed at monitoring the plant diversity patterns within the South African savanna. In particular, we were interested in which local versus large-scale factors determine the diversity, species richness. Sorry, no problem. Okay, thank you very much. Plant species cover, species composition of the South African savanna. Also, we were interested whether these factors differ between herbs or forbs and shrubs, as these two life forms of plants can be expected or presumed to be affected by different factors. Also, we were interested about the role of large herbivores in these diversity patterns, and last but not least, we also focused on the question which type of habitats or which type of sites host most of the typical savanna species that are of conservation concern. My colleagues have already introduced that, but even my data or the data that I'm presenting were collected within the Mosaic project, and therefore we had the same system of 60 plots that were arranged in a way that would account for 2 different bedrocks that occur within the Kruger National Park, and that would also differ in the water availability, basically by locating the plots close to perennial rivers, close to seasonal rivers, and on crests, which represent the open savanna. On each plot, we recorded all species of vascular plants, but we also allocated there the camera traps that would record the richness, I mean species richness and abundance of the animals that are present. Further, we also tested the effect of large-scale factors such as climate, fire history, character of vegetation, and intensity of disturbance. I will just go through this very briefly because this is sort of boring, but we used mixed effect models to test the effects of bedrock and water availability on species richness and diversity. Then we used ordination models which would test the effects on species composition, but these were also used to identify large-scale spatial factors that were potentially confounded with our research hypothesis. We used a variance partitioning procedure to separate the effects of the north-south gradient from the effect of Colophospermum opane cover. And we tested the effects of large-scale factors using regression models that would take into account the effect of local factors such as bedrock and water availability, but that would also include the significant spatial effects that have been identified by the ordination models. So let's start with the local factors. We found out that basalt, the communities or the vegetation on basalt is species poorer compared to the vegetation on granites. Concerning our habitats or differing in their water availability, we found out that the crests, which represent the open savannas, were species poorer or more poor in herbal diversity compared to both seasonal and perennial rivers. On the other hand, seasonal rivers were detected to be more species rich compared to both perennial rivers and crests in shrubs. And overall, we detected that out of the combination of habitats that we inspected, the crests on basalt were the most species-poor community that we worked in. On the other hand, crests reveal the largest share of typical savanna species that are likely to be of conservation concern. So crests are important from the conservation point of view. Also, crests show the highest cover of savanna grasses, differing significantly from perennial rivers and marginally significantly from seasonal rivers. Interestingly, we didn't find very convincing differences in species composition between basalts and granites. The herbal communities differed only marginally significantly on granites versus basalts, and the shrub communities did not differ significantly at all. Concerning the abundance of herbivores, which is also obviously a very important factor that impacts savanna plant diversity and species composition, we detected the largest abundance of herbivores on perennial rivers and the lowest abundance of herbivores on crests, which is sort of logical. If there is no water, why should there be herbivores? However, we detected a significant correlation between the herbivore abundance and the species richness of herbs within our study system. But the same relationship was, even though when you look at the graph, it suggests there is some positive relationship, it was not detected to be significant because of the high scatter. How to interpret this? This is sort of difficult because actually herbivores may increase herbal species richness. For example, via the mechanism of dominance control, where herbivores reduce the dominance of some plant or grass that would otherwise suppress other species. Or at the same time, the herbivores can increase microsite heterogeneity and therefore provide opportunity or provide niche for more herbal species. But also, of course, this is just a correlation, so it can be the opposite direction, that herbal species richness may increase herbivore abundance, for example, by providing a more stratified food supply. But at the same time, the fact that herbal richness and herbivore abundance are correlated doesn't mean that there is any causal relationship between these two measures. It can be just that both of these measures respond to some other factor that we didn't account for. Out of the large-scale factors that we were interested in, only temperature was detected to have a really strong, convincing, and consistent effect, negative effect on total species richness, or total plant species richness, which was even after accounting for the effects of local factors like bedrock and water availability, but also even after accounting for the large-scale spatial effects. Interestingly, we did not detect any convincing relationship, for example, for fire history, which is sort of counterintuitive. We however think that the effect of fire may be actually masked by the effect of bedrock, as we've detected that the vegetation on granites tends to be burned more often than the vegetation on granites. Also, it's sort of honest to admit that our tests are quite weak due to the limited number of replicates. Even though we had 60 plots, we actually had only 20 independent replicates because our plots were hierarchically arranged in triplets. And also, many of the factors that can be presumed to impact species diversity, like fire history, precipitation, disturbance, may actually not impact diversity or richness of plant species, but rather the composition of communities. We also focused on the effect of mopane, because mopane is often discussed and it's often presumed to suppress the diversity of plants, especially herbs, in the northern part of Kruger National Park. However, when we performed the variance partitioning to separate the effects of the north-south gradient and the effect of mopane, we found out that the effect of mopane on herbal richness and diversity was not significant. More importantly, this is in accordance with our field experience. We recorded several plots where the mopane cover, in the north of Kruger National Park, where the mopane cover was really, really high. And at the same time, there was a very high herbal diversity. On the contrary, we had some plots where there was very few or even no Mopane cover, and these plots were species poor. So, to sum it up, our results suggest that most of the plant diversity patterns within the Kruger National Park are determined by the local factors such as bedrock and water availability. And also, it's obvious that herbivores play a major role in determining the diversity patterns. However, we do not really know about the direction and about the causality between the herbivore abundance and plant species richness. We are quite convinced that plots on basalts are more species poor than plots on granite. What is the reason for that? It may be the stress associated with basaltic soils, because basaltic soils, when they desiccate, they sort of crack, and which may actually mechanically damage the roots of the plants, and it's quite understandable that many species cannot cope with that kind of mechanical disturbance and stress. On the other hand, it can be also due to the dominance of grass species. It was really apparent from our field experience that plots on basaltic crests representing the open basaltic savanna were really highly dominated by grass species. So it may be actually the grass species that are so dominant that they suppress the herbal diversity. Also, we were quite surprised that we did not detect many species that would be specific for either basaltic or granitic savanna. It actually seems that, and the results of the ordination models also suggest so, that the plant communities or the vegetation on basalt represents some kind of impoverished version of the communities on granites, without actually having too many specific species. With the exception of temperature, we did not detect any significant effects of the large-scale factors that we measured on the plant diversity patterns. However, I think it is sort of honest to admit that we are not really confident about these results, and we feel that our tests are weak, and the effects of large-scale factors are likely to be masked by the effects of local factors. Also, we do not think, and our results do not suggest that the effect of Mopane suppresses the vegetation in the north of Kruger National Park. It's when the effect of Mopane gets separated from the north-south gradient, the effect of Mopane is actually quite weak. I'd like to thank you for the attention. Sorry for taking too much time.
Thanks, Martin. You finished just on time. So if there's any questions, please grab him at lunch. So just before you go, I just have a couple of announcements, but I'd first just like to thank all the speakers in the session. Thanks for keeping to your time. I think you must have realized that I played quite a big part in putting the program together. So I'm not sure if you were scared that if you went over time, you wouldn't get a slot next year, but please don't worry about that. So just a few things. Jackie says, please, if you need an exit permit, you need to bring along your entrance permit so that she can do those for you. Your name tags, if you're not going to be these ones, if you don't want them anymore, please just drop them off with Jackie and she will use them again next year. We do still have some of these nice local community bags, so if anybody wants to buy another one, please see her. Your posters, please take them down at lunchtime if you want to keep them, because otherwise after lunch we're